Day 9. It's good. Everybody's been here and working hard, I assume, and having fun. I hope that, I really am looking forward to this whole debate process, and I'm hoping that people are putting good amounts of happy energy into that, because that will be a lot of fun. Jack, are you ready? Okay. Oh, does anybody have a power cord that fits an Asus computer here? That's right, we can show it from my computer. Actually, we can show your slides from my computer, probably.
Yeah, I just want to have movies.
So I want to yet once again, because we've had the honor of Jack's presence before, welcome Jack Gallant to the NeuroImaging Training Program. Jack is a professor of psychology at UC Berkeley, and he has made almost alarming new discoveries in brain science and brain mapping in the process of trying to understand encoding and decoding of our sensory features and our cognitive spaces. And his work has been very widely publicized. People will have seen that on TV and in Time Magazine, as well as in the journals. And he's a superb person, wonderful speaker, and almost ready to go, I think. You know, in the time that you've been doing it this morning, Vince Calhoun would have published 3 papers.
Isn't Vince an engineer?
Yes.
Have you ever read an engineering journal?
Oh, yeah.
There's a reason Vince can publish 3 papers. I like Vince. I do, he's really good. But engineering journals are not the same as neuroscience journals, trust me. Not even close. Engineering journals publish under the assumption that in 6 months, whatever you publish will be obsolete. And neuroscience journals publish under the assumption that people will be looking at this paper in 100 years. It's a very different attitude. And I'm in the IEEE, so I can say that. Okay, so in just a second here. Oh, it's up there. Oh, goody. Okay. So maybe this will just work. A miracle. Is there a laser pointer?
No, you're supposed to use your finger on your mouse pad so that people who are watching.
Oh. Oh, God, I hate that. Okay. The problem is I can't see it. Good morning. Sorry for the delay. I'm giving 2 talks apparently this morning. One is a talk on, it's a basic research talk. And this is basically the same as a talk I gave at MIT a couple months ago, which I sort of remember, but I've given variants of this talk a lot, so hopefully we'll manage to muddle through. And then the second part is going to be kind of a methodsy talk, but it's more going to be a philosophical issue about things that bug me. And since that's a really long list of things, I have a lot to talk about. I have a very different perspective than I can just talk louder. I'm just gearing up. Just give me more coffee, buddy. It's fine. All right. I'm just talking to rooms of 800 people. Mark wants me to yell. No problem. So yelling is another thing that I do really well. All right. I have a very different perspective on MRI than many people. MRI is kind of a weird field if you look over all of science because it's divided into kind of two camps. There are the developers who are either the MRI physics people or the software developers, and then there's the users who are largely either medical people or psychology people. And this is, you know, everybody kind of thinks this is a perfectly fine thing in MRI. This is not the way science used to work. It used to be that you had to have mastery over the whole pipeline from your instrument and building your instrument to collecting the data and publishing your paper. And that's, as technology has progressed, that's kind of broken down. It's broken down, I think, in MRI. And also the other place where it's starting to break down is in molecular biology. So, you know, when I was a kid and people would work for a year to clone one gene, everybody was doing everything by hand, and now you just buy a kit, or you use some machine that you buy from a company, and nobody really knows how they work. So I would encourage you to try to get mastery over as much of the pipeline as possible. If you're a developer, you should learn what the users actually do. And if you're a user, you should learn what the developers know, which means basically everybody should be taking an MRI physics class as the first prerequisite to doing MRI. I think if you can't code up your own pulse sequence, then there's something wrong. So the reason I have this kind of hardcore attitude is because I'm a neurophysiologist, and in neurophysiology, basically you have to do everything yourself because there's just not enough interest or money in it for a lot of development of technology that can be shared. So that's kind of how I learned the hardcore way, and that's kind of what I believe today. So I am interested in mapping things across the cerebral cortex, where things means whatever's there, right? The brain is just a big meaty computer. It computes stuff. There's a cortical computer subsystem that probably consists of, say, 200 to 400 distinct functional anatomical areas. And there are a bunch of subcortical giblets that are, of course, incredibly important, but which I ignore because there's just too many to remember, and I have a really bad memory. So I just ignore all that part, and I'm interested in cortical computation and cortical representation, basically what information is represented across the cerebral cortex. So the talk is going to follow this outline. I'm going to talk about mapping and then a particular experiment which you are probably at least partially familiar with at this point because it got a lot of press. And then I'm going to talk about a cool generative model that we have that unfortunately nobody cares about yet, but I'm hoping that will change. And then I'll talk about what good does it do me, which is a really common question we get. So we all know that you can do anatomy on human brains and parcellate the brain into its functional parts. Right? People have been doing this since the 19th century. By the beginning of the 20th century, there were several proposals for anatomical parcellation of the human brain. The one that everybody remembers is Brodmann, but there were several competing proposals that were all generally similar, but not exactly the same, that came out about that time, and just Brodmann kind of won the contest of history, and he's the one we remember most. Brodmann delineated something on the order of 100, areas or so, depending on there was an exact number, but it doesn't really matter at this point. Nobody doubts that these anatomically distinct brain regions of the cerebral cortex are probably functionally distinct, right? It would be kind of crazy if you had anatomically distinct areas that were not functionally distinct. So this is a first cut parcellation. But of course, Brodmann, like everybody else, is signal limited, right? If you only got a light microscope, there are going to be anatomical distinctions that you can't see, and so you should consider the Brodmann areas to be a lower bound on the true number of anatomically distinct areas across cortex. Now, more recently, we don't have to wait for somebody to die and then do light microscopy on their brain to try to determine anatomical parcellation. We can use this incredibly cool, but I would argue somewhat sketchy tool called diffusion MRI. Did we have lectures about diffusion MRI? Okay, did any of those lectures ever present error bars on the diffusion maps? Did they ever try to estimate the error on the maps? Did they ever try to tell you how accurate they were? Okay, they tried. Well, that's good. That's better than they used to do. So how convinced were you that they knew? Thank you. That's the correct answer. The diffusion imaging has become popular enough, and there's a really pressing medical need for diffusion imaging to work. It's become popular enough that there is now pressure to try to quantify diffusion imaging accuracy and figure out how well it works. And really, the only way to do that is to essentially go to an animal model and use an invasive method, slicing and dicing the brain and doing actual track tracing and anatomy, and then compare that to the diffusion imaging. When you do that, you find out that diffusion imaging, does an okay job on the big fiber tracks that you can, that are there pretty much in everybody, and it does a pretty poor job on small fiber tracks. It is really subject to distance bias, and once the fibers decussate into cortex, then of course all information is lost. So diffusion imaging, everybody wants to work. I also want it to work, but right now I just don't trust it. I just am very skeptical that it works in any way that can be replicated other than at the coarsest level. But there's a lot of work being done on it and a lot of money going into it, and I think there should be a lot of work going into it because we really do need it to work. So hopefully it will work sometime in the future. This is a pretty good diffusion imaging study, and as you can see, they proposed a 70-area parcellation. Now we know from Brodmann that there's going to be more than 70 areas, so we see that diffusion MRI in its current state is kind of underestimating the anatomical areas. Of course, there's structure and function that are going to be systematically linked together, and the reason for that is that on a short time scale, the brain's just a fixed set of wires, right? It takes about 20 minutes to grow a new synapse. So on a short time scale, all of your different thoughts have to flow over a fixed set of wires. You can up and down regulate a synapse pretty quickly, but you can't make a new synapse very quickly. So somehow or other, this anatomically stationary structure on a short time scale has a diversity of functional properties, and we don't really have much of any clue of how you will build a computer with these properties. But it should be the case that the structural information in the brain and the functional information in the brain are closely related and convergent and essentially tell you about the same thing, right? Ideally, it would be hard to imagine that wouldn't be true. So there's a lot of work being done on diffusion imaging by people who are better at that than I am, and I kind of ignore that, and I've focused instead on functional imaging. Now, functional imaging, of course, we all know has its own issues, right? Functional MRI isn't neuronal. It's a bold signal. It's an indirect reflection of neuronal responses. The bold signal is going to reflect both excitatory and inhibitory neurons. You can imagine if the number of synapses of excitatory and inhibitory neurons in the brain were equal and the brain was balanced in its activity, as is occasionally proposed, then there wouldn't be any MRI signal at all. But fortunately, about 80% of the neurons in the mammalian brain are excitatory. So you can try to do parcellation with functional methods instead of diffusion methods, and this is a pretty good study of using resting state MRI. Did somebody talk about resting state MRI? Okay, so what was their X variable? I mean, the Y variable was the bold activity in the brain. What was their X variable? Did they tell you? There is no X variable in resting state MRI, right? It's just Y's, which is, so when you have a regression problem, all of science is a regression problem, right? I manipulate something, and I observe something, and I build a model that relates what I manipulated to what I observed, what I measured. And in resting state, you didn't manipulate anything. You only have your Y variables. So since you don't have any X's, you can't actually solve the regression problem, so the only thing you can do is look at the covariances of the Y's, and that's what everybody does in resting state. You look at the covariances or the correlations between the Y variables and try to apply cluster analysis of the correlations to divide the brain into its parts. That might work. It's kind of the only thing you can do with resting state data, and that's what people do. And it does, in fact, I'm sure you saw in the resting state lecture, resting state does give you parcellations that are generally consistent with large-scale functional parcellations. In fact, way more than I would have expected. If somebody had told me they were going to do this experiment, 20 years ago before anybody did it, I would have said they were insane and this would never work. So that was obviously wrong. This does work. It's just unclear how well it works and how consistent it is. But you can get a large solution of many area parcellations from resting state data if you try. The different groups that do this have different algorithms for clustering the covariances, and so they end up with slightly different solutions, and they haven't really converged yet, but there's so much resting state data being collected that will probably happen eventually, I would assume. I would say people do resting state studies because they seem easy. And everybody, I'm just as lazy as the next person. Everybody's lazy, everybody wants a free lunch. And in resting state, it seems like you're getting a free lunch. Oh, I just put something in the magnet, get all this data, and everything works great. So, but of course, in science, there's really no free lunch. So really, my only complaint about resting state, my real core complaint, is that nobody calls it functional covariances, they call it functional connectivity. A word coined by Carl Friston, and I wish he had never done it because everybody picked up on it and everybody talks about functional connectivity, and this has nothing whatsoever to do with connectivity. That's an assumption. This is about correlations. And they might reflect connectivity and they might not reflect connectivity. So when I talk about resting state, I'll usually talk about functional covariances, which makes it hard to publish papers because nobody knows what you're talking about. All right. Now, you can also, of course, use the classic method for trying to parcellate the brain. This is a really nice review paper by Kathy Price, who looked over about 900 studies of language, and these were almost all standard contrast studies, right, using the standard subtractive factors logic for MRI that Nancy Kamwisher and others developed in the mid-90s, where you have two different conditions and you subtract them and you look at the difference and you try to use that to identify functional areas. And if you then do a meta-analysis of a lot of different studies, you can find general patterns that seem to be somewhat consistent. But every meta-analysis of every function, except maybe vision, you could argue, because vision's of course a much easier problem than every other part of the brain, every meta-analysis of every other cognitive function ends up with parcellations, functional parcellations that are much coarser than we expect the anatomy should be, right? The anatomy suggests that there's going to be something like 200, maybe 200 to 400 anatomically and functionally distinct areas, and all these meta-analyses of classic MRI studies using functional contrast end up with far, far fewer areas. So these are lossy. There's a lot of type 2 error, right? There's stuff going on in the brain that we can't see using this approach. Okay, so when we started working on functional parcellation, we had a set of goals that we were trying to achieve. We wanted to obtain a functional parcellation for real stimuli and natural responses. And the reason for that was we wanted to make sure that our results were going to generalize to the natural world. We wanted to look at as broad a range of stimuli as possible. We wanted to uncover as much of the intrinsic tuning space as possible. We wanted to account for individual differences in the parcellation. That turns out to be a really hard problem because, of course, looking at individual differences is kind of a second-order problem. First-order problem is looking at the group mean. Second-order problem is looking at individual differences. You need a lot more signal to look at individual differences than to look at the group mean. We wanted to look at, for changes of functional parcellation with task demands, meaning attention, learning, things like that. And we wanted to link the functional parcellation to the anatomical parcellation. And I would say we've done all of these things in the development in our lab except for this last one, which we really haven't put a lot of work into yet. So I come from MRI. I come to MRI from the back end. Most people come to MRI from the point of view of psychology, and they're interested in psychological theories and how they're implemented in the brain hardware. And I, since I'm a neurophysiologist, I tend to think of the brain hardware first and trying to extract the function of the brain hardware directly from the brain rather than coming at it from a theory. I'm a very sort of data-driven kind of person, and that's simply because I have certain biases which you've already detected. One is the first law of neuroscience is the brain is complicated. Second law of neuroscience is no matter what you think about the brain, it's probably wrong. And the third is the brain really doesn't care what you think about the brain. So those kind of assumptions make it difficult to come up with a theory that you can test that will actually generalize well in the real world. And I would say that oftentimes in psychology, I'm in a psychology department, so I have a lot of debates with people who complain, well, you have to start with a theory, otherwise you don't get anywhere. And my response is, starting with a theory is great if it's a theory that's going to work. So if you think your theory is correct, put it on a robot and let's see if the robot behaves. And as you notice, we don't have any robotic overlords yet, because every psychology theory that's ever been put on a computer or a robot basically fails in the real world. And when we do have robotic overlords, they will have been trained not using a theory, but using some machine learning method and big data, right? All the theories we have about real cognitive function are very, very brittle, and they work under a very narrow range of circumstances, and they break really, really fast in the real world. So the problem is, if your theory breaks, what do? Right? It's not obvious what you do, because essentially that means you're stuck in a local minimum, and you don't know what the global minimum is. So I tend to do things from a data-driven point of view. So in that case, if you're taking a data-driven perspective, then the goal is just to map as much as you can about cortex. And so it's useful to think about how cortex is mapped. So on the bottom here, on the left, we have a retinotopic mapping stimulus, and on the bottom we have the primary visual cortex of an animal that was injected with a radioactive dye, and the radioactive dye was bound to glucose, and when the neurons fired, the neurons took up the radioactive glucose, and then the brain was extracted, flattened between two pieces of glass, like a piece of photographic film. And now you can see the retinotopic map here on the lower left. And if you look, this is the global map in primary visual cortex, is of course retinotopy, right? Because there's many retinotopically organized visual areas. But if you zoom in just to one little spot, like this little spot here, and you do, say, if you could, do some sort of intrinsic voltage sensitive dye or something like that, some functional imaging, you would see that there are orientation columns, and there are spatial frequency columns, and there are ocular dominance columns. In fact, there are about 10 to 15 different variables that are represented at each retinotopic location. So primary visual cortex is a global organization that's retinotopic, and a local organization of these other like 10 or 12 dimensions that are all compressed and projected on each individual retinotopic location. So there's a global scale and a local scale of organization. And of course, ideally, you'd like to recover the entire tuning space, both the global information and the local information from each brain area. So you need some method that's going to find the areas, and then within each area, extract the global maps and the local information. If you want to solve everything, you may not be able to do that. Okay, so I'm going to demonstrate how we solve this problem with a little story about an experiment that we published last April, which was the simplest experiment we've ever done. People just slide in the magnet, and they listen to the moth radio hour. So I don't know how many of you know the moth radio hour. It's basically stand-up storytelling. It's a contest among the storytellers to tell the most compelling, interesting story to the audience. So these stories tend to be autobiographical. They're about, love and death and loss and redemption and regret and all those human kind of things. So this is in some sense a biased set of stimuli because they're all autobiographical stories. So the results we get from this experiment may not generalize to other kinds of narrative, but we wanted to start with this because one thing that Uri Hassan has shown is that these moth stories are very, very compelling, and they produce a lot of brain activity. So subjects actually love being in this MRI experiment. How many people have been in an MRI experiment? I'm just kind of curious. Okay, how many people liked being in the MRI experiment? Wow, okay, so a few sick and twisted people, you should all become engineers. And the rest of you, you would actually like this experiment because it's really actually very easy. You just lie there, you listen to these stories. You don't have to fixate, so you can close your eyes. It's staying awake's not a problem because the stories are great. This is the one experiment we've done where we didn't have to pay the subjects to be in the experiment. We did, but we wouldn't have had to. People would have been happy to do it. So, okay, so you listen to a story. You're lying in the magnet listening to a story. Well, what goes on in your brain? Well, anything that you can think about this story has to be represented somewhere in the brain, right? The sound spectrogram is represented at the cochlea. The phonemes have to get extracted from the story. The articulatory features you might be represented somewhere in the brain. The syntax, the semantics, all of that information has to be represented somewhere in the brain. So if we had the ultimate brain neuroimaging device, we could essentially reconstruct the representations of all those different kinds of information simultaneously from brain activity. Of course, we don't really have that kind of neuroimaging device. We've got an MRI machine that, is better than anything else out there, but really kind of sucks. So the biggest problem with MRI, of course, is the slow time course. There's really not much information in these hemodynamic signals below the level of about 0.3 hertz, 3 seconds. So anything that happens at a scale faster than 3 seconds, you're not really going to be able to see. So you're not going to be able to see any kind of sound spectrogram stuff. Phonemes are going to be really hard to extract from these data. But any processing that has to do with long time scale things like the emotional content of the story or the semantic content of the story or the narrative, you should be able to pull that out. And you might be able to pull out some information that's related to syntax, because that occurs at a variety of time scales, some of which are within the time range that we can measure in the MRI machine. So like every other brain imaging device, this experiment, this is going to be lossy. It's going to be mostly type 2 error, right? Most of the stuff in the brain that's associated with processing these stories we're not going to be able to see. All right, so now we have to come up with a way to analyze the data. After all, we've got some X variables, which are some stories, and we've got some Y variables, which are some brain data. How do we process this data? We don't have a theory about what should be represented where or how. So we have to do this in a data-driven manner. So the way we do it is called voxel-wise modeling. It's a pretty straightforward process, but there are very few labs that do this. My lab does it. Tom Mitchell's lab does it. Bertrand Thierion's done a little bit of it. Most people do not do this. Really, this is just the general linear model, right? This is just regression. We're going to take each individual voxel, and we're going to regress it against some transformation of the stimulus, and we're going to try to figure out from that process which features in the stimuli are represented in each voxel. So this is a multi-stage process. First, we hold the brain data out. We take the individual stories, and we break them up into a bunch of different feature spaces. So these are nonlinear, usually, transformations into some feature space that reflects the stories. We might have a bunch of spectral features, which are just going to be time by frequency. For a typical story, you might use 500 of those. You might have, that probably says phonemic and articulatory features. I can't really tell from this angle. So that's going to be, there's about 60 phonemes or articulatory features in English, so we can extract all the phonemes from the stories. Then we might have some sort of syntactic model. Now, extracting syntax from stories is really hard. You might think, oh, you want to use a Chomskyan deep grammar. But if you try to take, say, a moth story and build a Chomskyan deep grammar graph for that story, it'll take you about 20 years, and the team of 50 linguists you have doing it won't agree at the end of the 20 years. So you pretty much have to use some data-driven method to do this. You can use a neural network to extract the syntactic features, or you can use some sort of, say, hierarchical hidden Markov model. You might end up with about, say, 40 semantic features, syntactic features. And then you have semantics. Well, how do you represent semantics? Well, semantics is the meaning of the story. So how do you represent meaning? After all, these are just words. You have to represent meaning somehow or other. And the normal way you represent meaning in natural language processing computer science is you represent the meaning of each word in terms of its associations with other words, its correlations with other words. So, for example, dog and bite are highly correlated words, but dog and Ferrari are poorly correlated words. So we can take each individual word and we can look at its correlations in a huge corpus of words, and we can represent the meaning of the word in terms of its covariance with these other words. So in this particular experiment, we built a 985 long semantic space by taking the 985 most common words in the English language. These are the most common nouns and verbs, not pronouns and things like that. And we find the covariances of those words with 6 billion words in a giant corpus of text. So now we have essentially the meaning of these 985 words in terms of their covariances with each other and with these other words. And now we take the new words in the story and we project them into this semantic space we've created. So whatever the words were in the story are going to be modeled as their covariances in a large corpus of text with these 985 words. So we're essentially projecting them onto these 985 words. So if a word down in the semantic space is dog and a word in the story is wolf, then wolf will project very highly onto the semantic concept of dog. And if another word in the semantic space is car, then wolf will project very low. It'll have a very low weight projecting onto car, right? So you can kind of see how that works. Okay. So now we've got, so now we haven't done anything with the data yet, right? All this is just taking our words and projecting them into these semantic spaces. You can think of each of these semantic spaces as a hypothesis about some kinds of features that might be represented somewhere in the brain from this story, right? So this is 4 different hypotheses we have about what the brain could represent in this story. Okay? Now, we take our brain data, and for each of these, say, 50,000 voxels in cortex, we're now going to use regression to map each of these features onto the bold responses that we actually measure from that voxel. So you can tell that there's going to be something on the order of, I don't know, maybe 2,000 features here, and they'll all be fit simultaneously. So for each individual voxel, we're going to take these 2,000 features. We're going to use ridge regression. It's MRI data, so even if it's not supposed to be Gaussian, remember the first thing you do with MRI data is you make it Gaussian, right? MRI has signal and noise, and no one knows what the noise is. So rather than doing the intelligent thing, which is estimating the noise, what everybody does is they just take all the Gaussian stuff and call that signal, and everything else they call noise, and declare victory that way. So we know that this data are going to be Gaussian because we made it Gaussian. So since it's Gaussian, we can use a standard GLM regression method like ridge regression. So now we're going to take each of these features, we're going to regress them onto the bold responses, and we're going to try to predict the bold responses in the estimation data set. And this is going to take about, say, 2 hours of data. Now, in the second stage of the experiment, and this is the most important part of the experiment, and this is what almost no one in MRI does, and to my eternal regret, we take a separate set of stories, okay? And we now take those stories, we project them into these feature spaces, and now we take the models that we fit in the last stage, we multiply each of the features by the weights we fit in our regression equation, and we get a prediction. And we compare the predicted response to the observed response in the second set of stories. So, you notice I didn't say anything about statistical significance here. I talked about prediction. And there's a very good reason for that. I think statistical significance is an abomination on the face of science, and it was a really bad idea. And it was great in 1930 when Sir Ronald Fisher came up with it because nobody was doing anything, but now it should die a very fast death, and we should focus on prediction. And if we only used one set of data, go back. If we only used one set of data and we fit our model to our data and then we predicted that same data, well, we could arbitrarily overfit to this data and we could get basically perfect predictions, right? If I have 2,000 features and say 500 time points, I'm going to be able to predict every time point almost perfectly. So I can't use the same, if I'm looking at predictions, I can't use the same data for estimation of my model and validation of my model. So I need a different set of data to validate my model. So in this stage, all I'm doing is taking the models I fit in the last stage and looking at the predictions, and I'm comparing the observed to the predicted. Now, there are some subtleties involved in this process of prediction, because you can imagine if I only took one bold response and I try to predict that bold response, even if I have a perfect model, I will not predict that bold response. Why? Because that bold response has noise in it, and so it's a sample from a distribution of potential bold responses that I could have recorded. So the perfect prediction will still not actually give me a correlation of 1 with my observed variable. So I have to calculate something called a noise ceiling. And I can do that based on an objective estimate of the noise in my validation data. I can say, well, given that I have, say, 3 repetitions of this validation data, I can calculate the standard error of my distribution, and that tells me how far the true mean is from the sample mean, and that tells me how good the best possible model I could ever get is. So if the best possible model I could ever get is predicting 80% of the variance, and my prediction predicts 70% of the variance, I now know exactly how close I am to solving the brain. I am 7/8 close. You will never get that from significance, right? Because statistical significance only tells you, is my data likely to be random? And it really doesn't even tell you that. So it has nothing to do with prediction. It has nothing to do with importance. It has nothing to do with effect size. Prediction is the ultimate measure of effect size. So we always do predictions. And if I was a ruler of the universe, the first rule I would impose is every journal has to require reports of effect size. And even if you don't do it my way, if you just compute effect size another way, the world would still be a much better place. So now let's look at low-level features. So This is really hard because I really can't see this thing at all. So here are 3 feature spaces, an articulation space, a spectral space, and does that say semantic over there? God, I should move over here. Oh, but then I can't. All right, I'm constrained. Okay, so this basically just shows the predictions of these 3 models across flattened cortex. So I don't know, how many people are used to looking at flattened brains? That's about what I would expect. Okay, everybody should look at flattened brains. I grew up in the David Van Essen lab, and if you grew up in the David Van Essen lab, you learn to look at flattened brains, whether you like it or not. And eventually, if you do that long enough, it's like any kind of marine training exercise. Eventually, if you do it long enough, you learn to love it. So I tend to make flattened brains of everything. They do have some advantages. You can see everything at once, right? The disadvantage is the relationship of different parts of the brain to the sulci and gyri that you know so well is now completely abolished. So you just kind of have to memorize where the parts are based on knowing the orientation of the flattened brain. So in this particular flattened brain, this is the visual system in the middle here. This is prefrontal cortex left and right, at the left and right. This is auditory cortex down here, sort of at the bottom middle. This is the motor and somatosensory strip here and here, right? And so this is going to be the precuneus folded inside, and this is going to be the temporal lobe down here somewhere, right? So that kind of gives you an orientation. So now we've just plotting predictions using a color scale, and you can see that the spectral model works pretty well in auditory cortex. And that's exactly where you'd expect it to work well. The articulation model works well in secondary. This is a higher order auditory cortex. This white outline here is auditory cortex. This is a very generous auditory cortex localizer. So it's including both primary and multiple secondary auditory cortices. So the articulatory model fits well in sort of higher order auditory cortex, particularly in this subject anyway, in the left hemisphere, and not really all that well everywhere else. And this semantic model fits well almost everywhere, right? So if you look at this data, your first thought, if you're the person running this experiment, is to pay attention to the semantic model, because that seems to be where most of your signal to noise is. And after all, you know, if you have this open-ended kind of experiment where you're not trying to test a specific hypothesis, paying attention to where you have the most signal is a smart move. But we try to look at this whole thing. So one of the things you can do is do something called variance partitioning. And this deals with the collinearity of these models, right? The spectral model, the phonemic model, the articulatory model, syntax and semantics are all somewhat collinear. So if you just fit all the models independently, you don't really have a good idea of each model's individual contribution to the data. So in psychology, we call this variance partitioning process the part and partial correlations. And in other areas of science, they might use part and partial correlations, but you can just think of it as a variance partitioning problem. It's just a set theoretic approach. So we have 3 models in this case, and we can look at the independent contributions of these 3 models, and we can look at the joint contributions of these 3 models. And it's not important for today's talk. I'm just putting it out there as something you can do. And when you're testing multiple models with natural stimuli, they're almost always collinear, so you almost always end up having to do some sort of variance partitioning. And we published a paper on variance partitioning in Vision last fall, if you're curious to look at it. You can zoom in and look at how these various models are represented on a little tiny patch of cortex. So here we're just looking in the superior temporal gyrus, and you can see the normal features of auditory processing that people have mentioned before. This doesn't have the frequency tonotopy map on it, but you can get tonotopy out of this experiment as well. So the person who was in charge of this whole project was Alex Huth, this really awesome graduate student, now postdoc of mine. And Alex looked at this data and said, well, wait, most of the signal here is in semantics, so I want to look at semantics. So, sorry, I flipped too far here. Come on, computer. Okay. So he decided to look at semantics, and the first thing he did was look at all the previous studies that have been published on semantics. There have been a lot of studies of the meaning of language that have been published over the years, almost all of them, virtually all of them, using localizer methods. And therefore, they're likely to be lossy. And there have been a few good review articles that have been published the last few years. This is a pretty good one from Jeff Binder showing the 2011, does that say 2011 or 2009, view of semantic organization throughout the brain in the case of language. And you see the usual course divisions into different classes of semantic. It's much easier to give these talks when you can actually see the slide. Oh, wait. I know. It's not only where I'm standing, it's also when I'm running out of time. Sorry, I have a bad case of cold, so I need prosthetics. Oh, this is much better. There are words on these slides. All right. Yeah, so here we have modality-specific processing, temporal-parietal convergence zones, semantic memory interface. These are all kind of labeled, presumed functions from various semantic experiments. Now, the first thing when you do voxel-wise modeling, the very first thing you do is you look at predictions. This is before you interpret your model, right? Remember, science has 3 stages: observe some data, make an explanation, hopefully a quantitative explanation in terms of a model whose dimensionality is lower than the data you started with, and then predict, right? And so if your predictions are lousy, nothing in that model explanation part is going to matter, right? What difference does it make if you have a model that only explains 0.5% of variance in your data? That would be silly. So the very first thing you do when you run this pipeline, since you have the ability to get predictions, is you evaluate the predictions, because you want to only interpret parts of the brain where you have good predictions. It doesn't do you any good to try to interpret the model in places where the predictions are poor. So you can see that the semantic model predicts really well in a lot of different places in the brain. It doesn't predict particularly well in auditory cortex or in the somatosensory motor strip, or in primary visual cortex and secondary visual cortex. But it does a good job posterior to auditory cortex, in the temporal parietal junction here, in the retrosplenial cortex, and Broca's area will be somewhere around here, and all over prefrontal cortex. So there are a lot of locations where the semantic model predicts well, and that suggests that these parts of the brain represent semantic information in the stories. Okay, so now I have a problem. It's a good problem. It's not one you usually think about. I have too much data. So this is 985 features. This is the wrong slide. In this experiment, there were actually about, oh no, this is the correct slide. There were about 1,000 semantic features in this experiment. And so if each subject, we have 1,000 semantic features and say 60,000 voxels, then we have a matrix of weights for each subject, right? And in our experiment, we have, say, 7 subjects. So we have a huge amount of data that we have to interpret. And that's a problem. This is too much data to interpret. So in science, when you have too much data, you always do some form of dimensionality reduction. In psychology, there are a lot of ways to do dimensionality reduction that come under the various names factor analysis. There are 1 million forms of factor analysis that you can do. Engineers usually just do principal components analysis or independent components analysis because they don't have such a need to do dimensionality reduction. So if you're trying to distribute your results to the widest possible audience, the first thing you do is PCA, because it's the dumbest form of dimensionality reduction, and it's the one that everybody will know. And if they don't know what principal components is, they won't admit it, so that's okay too. So, so generally, we always start with principal components analysis, then we do some more complicated things and decide whether, whether we need to do it. And in this particular case, most of the story was told successfully with principal components analysis, so we stuck with that. So if you do PCA on these individual data matrices, you find out that it takes about 60 principal components to account for 95% of the data. And that's, and so that tells you really there aren't, we're not recovering 1,000 semantic features from these data. We're recovering something like 60 semantic features, right? And so now you can look at the semantic features for each of the individual subjects, and you can essentially correlate them, because after all, a principal component space is just a vector space. So subject A has a vector space and subject B has a vector space, and I can look to see how well these correspond to one another, how well they align. And if you do this, you find out that the first 4 to 5 principal components are almost exactly the same between the subjects in the experiment, and they're not correlated with the stimulus principal components. If they were all just the stimulus principal components, then we wouldn't actually have any brain data. We just have an artifact of the stimulus, so we have to check that. Now, I should mention that all of these subjects are graduate students and postdocs in my lab, so they all are similar in the sense that they're crazy enough to be in my lab, and they're educated, kind of computationally savvy people, but they did come from quite many different places. They came from all over the US, a few came from Europe. The Europeans, we made sure that they were reasonably bilingual before we use them. But we've actually run this on people who were late bilingual learners, and it works very, very similarly. So you might argue that our subjects are biased, but for this biased sample of subjects, this works well. So now we have a principle. So now we can put all the data together. We just basically take these 7 matrices and we concatenate them together, and we do principal components on the whole thing. And we take the first 4 PCs, and that gives us a common principal component space across all of the subjects. So now, what is this principal component space? Well, remember, the principal components is a reduction of the data matrix. And the data matrix is simply a matrix that maps the stimuli onto the responses, right? So the principal components is just a subspace, a 4 or 5-dimensional subspace that maps the stimuli onto the responses. So a very interesting thing to do when you have this dimensionally reduced data is to try to interpret both the stimuli and the responses by looking to see where they project into this principal component space. So for example, if you take the individual words, the 985, not the words, the semantic concepts, the 985 semantic concepts, and project them into the principal component space, you'll end up with like a 3 or 4-dimensional space. So if you then color the words according to their loading on the 3rd PC, and you align them or array them according to their locations on the first 2 PCs, you get kind of a 3-dimensional map of the semantic concepts. You can also take each individual voxel and you can project into the semantic space. And remember, in both of these cases, we're only looking at the first 3 dimensions of the semantic space, and we're coloring the 3 dimensions red, green, and blue. So the color, the difference between colors of 2 semantic concepts or 2 voxels tells you how far apart the concepts are or the voxels are in this semantic space. So here is PC1, PC2, and PC2 and PC3, and we're just looking at the semantic concepts here. Remember, we're collapsing over all of the voxels in the brain. So you can think of this as telling you how the brain, what semantic concepts the brain views as similar, and what concepts the brain views as different when you collapse over the entire brain. And if you look down here at the lower right, you see concepts such as family things, parent, child, wife, daughter, and bad things that happen to families, like murder, divorce, confession, and things like that. And then up here, if you look up at the upper left, you see material descriptions of objects like thick, thinner, metal, green, so on. So this is, you can see the main axis of variation in this data goes from upper left to lower right. So that means if you wanted to make a localizer that would most discriminate between different types of semantic concepts in the brain, you would pick material properties of objects as one of your localizer conditions, and bad things that happen to families as your other localizer condition. Right? You can see this is a continuous mapping space here, right? We have on the lower left here, we have time and number and date and places. Those things go together. On the lower right, we have families and bad things that happen to families. And we have abstract concepts down here too, like thinking and dreaming and so on. And these are various kinds of things related to nouns, their shapes, their sizes, the actual objects, and their material properties. But this isn't really what we set out to look at. I mean, this is from a linguistics point of view, this may be interesting, but from a brain point of view, this isn't interesting because we've marginalized over the whole brain. And the whole point was to find out how semantic information is reflected in the brain. So what we want to do is we want to take all of the individual voxels and project them into the same semantic space. And that's shown here. So this is a flat map of one brain. Again, left and right hemisphere, visual system in the middle, prefrontal cortex at the right and left, precuneus here at the top, and auditory cortex sort of down here in the middle. And On the top here, I'm showing you the semantic space, but instead of showing you all 985 individual words, we did a cluster analysis on the semantic space and pulled out something, I can't remember exactly, on the order of 12 clusters, and just put those labels on here so you can kind of think about what this color map means. Because the color map used at the top, this is using the 3PC colors RGB, and the color map in the bottom, which uses the 3PC colors RGB, are the same. Right? So red locations on the flattened cortex here are locations that represent social concepts. Green locations are cortical locations that represent material properties of objects. Blue, as I recall, is maybe places. Can't really remember offhand. So you can see that this map is complicated and rich. And it's much more complicated than that Binder meta-analysis that I showed you several slides ago. These maps have a lot of interesting structure. You see that in temporal parietal junction, there's this big red spot, and it seemed to be surrounded by this green penumbra. Down here in lower ventral visual cortex, we see material properties of objects represented. Higher order auditory cortex seems to be social information. And then if we look here in prefrontal cortex, there's a lot of really rich structure up here, including weird repeating motifs. Like you see here, there's a green, pink, red, green, pink, red, green, pink, red, green. You know, and these are very complicated. Of course, I'm just showing you a picture here. I haven't quantified anything. I've weighted the individual voxels by their prediction values, so you know that this data is reliable, right? It predicts well because we're doing a completely honest cross-validated prediction. But this is just one subject. We don't really know how much of this is, say, consistent across subjects, how much of it may be due to stimulus bias or anything like that. We have to do more analysis for that. So one thing you can do is look at, hang on a second, make my life a little easier. So before I show you how to further analyze this data, let's do a little demo. Do I have sound up here? Oh, I've got it. Sorry about that, guys. Okay, so I don't know how loud this is going to be, so you should.
This looks like an onion, he said, holding up one of the bulbs.
Okay, so this is a little demo of what these
data look like. He made holes with the dibber and set the bulbs inside, and he was careful when he patted down the dirt so they wouldn't turn.
On the right, you can kind of
Just
see
wait,
a karaoke view.
You'll see in April how it works. The plant was inside.
A beechwood peacock with
This is a validation data set where the stories have been cut up rather than just being one story.
Colors.
And we flattened out the cortex here. And now we are painting the cortex with the activity as it comes in while people are listening to the stories. We've adjusted for the time delay. And we've colored the individual voxels according to their semantic activity.
Christmas and Hanukkah and Ramadan and all pagan festivals of that season of winter.
What a crazy world we're bringing our children into. He thought it sounded like the kind of statement that brings people closer together, pointing as it did to their common fate. But the sexy mom just glared at him and took the healthy living supplement, too, without asking.
He put Lily in charge of the party while he was gone, and then he walked downstairs. And there must have been 5,000 people milling around, wrapped in furs or long overcoats or ski parkas, leather jackets, high school and college kids, heavily champagne 60-year-olds, linking arms and singing.
Only the two front windows with white shades lowered were not somehow blanketed. Your eye was constantly drawn to where the material
hopefully
converged
you can see that the
mid-ceiling,
locations
by a
of
dazzling
activity
pink spotlight
and
that
the
looked
semantic
like it might have
loading
just
of the
vaporized a
activity
flamingo.
changes dramatically across these stories. Right? When the stories are social, you tend to see sort of more prefrontal activity and more parts of the brain that appear to be representing social concepts. When there was that story about the windows being broken in a house, you see activity in the parts of the brain that are representing information about material properties of objects. So the dynamics
And you
of
shall
these
remember that I am that I am.
of these responses are, reasonably consistent with the model. Now, one artifact you might think of is, well, wait, maybe this is just some strange property. Maybe these results are just some strange property of listening to stories. Maybe you get something very different if you read stories, and most MRI experiments don't use listening. Did anybody talk about auditory MRI in this class yet? No? So usually, most people who are doing auditory MRI, they use a dead time procedure, where there will be a long period of silence during which the sound is played, and then immediately after the sound is played, you start recording your MRI data. And then you record that for a few seconds, and then you have a delay period again with silence, and then you play your sound, and then you collect your MRI data. In this experiment, we found in piloting that, first of all, the stories didn't make sense, or they weren't as compelling when we broke them up that way. But also, we found that you quickly learn to stream these stories. So when you're in the magnet listening to the moth stories, while the magnet is actually running, it's as if there are two different tracks playing. And you can pay attention to the stories and just forget the magnets on. So we ran these stories while the magnet was running. And as you know, the magnet is very loud. So we were somewhat concerned, and the reviewers were somewhat concerned, that maybe this is an artifact of the way we collected our data. So one way around this is you can present a reading experiment. And the way Fatma did this experiment is she calculated the time of the onset of each of the words, and she basically made an RSVP stimulus where the words appear on the screen at the exact same times as they appeared they were spoken in the stories. So when you're watching this stream of single words appearing in the middle of the screen, you can follow the stories quite well. Some of the small words, the pronouns and words like is and the and things like that will get masked, but you can follow the story really, really well. And so now you can fit a semantic model to both the listening data and the reading data on the same subject. And if you look at these maps, they're almost identical. There are some slight differences in emphasis that may have to do with attending differently in the two situations. Like, notice this green is lighter than this green, and this red is a little more reddish than this red. So it may be that, in the story, people are emoting, right? There's emotional, expressive information in the stories that you don't really get from reading, and this map could be slightly biased by that. That was another concern we had. But in general, the semantic selectivity of this map and this map are almost identical, including, you remember I talked about these weird little green and pink sort of counterpoint patches in the prefrontal cortex? You see that exact same thing in this subject down here. So even the fine details of this subject's data look the same for reading and listening, and in fact, that's true for all our subjects. So that doesn't seem to matter. The only thing that's different is activity in higher order visual cortex, as you'd expect, because in one case there's words on the screen, and in the other case there isn't. Now there are other things you could look at. You could look at, well, prosody we've taken care of because there's no prosody in the reading experiment, so we don't have to worry about that. But there is narrative information in these stories, and we have not coded these stories for narrative information yet, and so we have not built a narrative model. There is one study I know of that uses a very similar method. It's kind of a reduced, simplified voxel modeling method from Spear and co-authors from a few years ago that, showed that it should be possible to recover narrative information. But we haven't pursued this yet because we haven't settled on a narrative model. Narrative is one of these areas of science that no one agrees on, and there are no good natural language processing computer science models for narrative yet. So we're kind of waiting until there's an automated machine learning NLP way to process narrative, and then we'll probably attack this problem. I should mention that the functional representation depends on task. I mentioned attention a couple slides ago, and there's a paper we published in 2013 showing that semantic selectivity for movies varies quite markedly depending on the task you're performing. So in this particular experiment, people were watching movies, and in one condition we asked them to search for vehicles, and in another condition we asked them to search for cars. This is the semantic map you get when they're not searching for anything. In this particular map, green means people, and red means scenes and cars. I know it's the opposite of what Alex did, but don't worry about that. And you can see if we look at the active viewing situation, When people are searching for humans, the map becomes much more green, and when they're searching for vehicles, the map becomes much more reddish blue, and that's completely consistent with the task they are performing. You also see these changes in individual single voxels. So one really nice thing I should just, as long as I'm advertising for voxel-wise modeling, one nice thing about voxel-wise modeling is you get all your data in single subjects. You don't have to average over subjects. Every single subject counts as a complete data set that you can analyze on that subject's own. So you can pilot your experiment in one subject because the signal to noise is so much higher than the conventional way of doing things. The second thing is you can look at, say, tuning curves for individual voxels. I didn't show you that in the previous slide, but it's shown here. This is the attentional tuning curve. This is a word net graph here, so it's a giant tree when people are searching for humans versus searching for vehicles, and you can actually see the tuning shifting in a single voxel. Yes? How much data is needed for each subject?
Code available on website?
The eternal question. I'll answer both those. So A, how much data is needed for each subject? That depends on the experiment. So this attention experiment that we published in 2013, it was 1 hour of data per subject. Alex's experiment on the moth stories was 2 hours of data per subject. The movie experiment we published in 2011, where we did that motion movie reconstruction, probably everybody's seen at this point, that was 3 hours of data per subject. So the general way we do things in the lab is you design a new experiment, and then you're going to pilot this experiment. And so you do some piloting to determine what are the appropriate task parameters, how exactly do you want to structure the experiment. And then once you've decided on that, you collect way more data than any sane person would collect on one subject. So for example, on the moth stories, you might collect 5 hours of data on the moth stories, okay? So now you have more data than you are ever actually going to collect in the experiment. But you can start chopping away data on that one subject to find out where is the cusp where your model quality falls off dramatically. So I think in the movie experiment we published in 2011, I think Shinji Nishimoto was in the magnet watching movies for something like 9 hours before we did the experiment. And then he determined that he only needed 3 hours of data. So all the other subjects were run with 3 hours of data. And his data was chopped down to 3 hours. And then I think Alex was probably in there for like 4 hours. I don't actually remember offhand. And then he decided he only needed 2 hours of data. So that's how we do it. And the nice thing is because this is a single this is essentially a single subject design, right? Just like psychophysics. You can do all your piloting with one subject. Right? Obviously, if you're looking at individual differences, that's not going to work. But for just figuring out where your signal is and what your task parameters should be, it's really handy only having to do one subject. Because in the normal, way of doing MRI is very, very inefficient and expensive. Yes. Go ahead. There's no. How long can I talk till 5 a.m. tomorrow morning, Mark? When is my time? There'll be a break for the speaker. Okay. Go ahead.
Okay, I was just wondering when you compare listening versus reading, did you look at replication in single subjects, like just one modality, like just listening and replication across time? So because I was just wondering, are these changes you see there, or these differences really due to listening versus reading, or could some of the variance be due to changes that occur over time in a single subject?
Okay. Those are two good questions, or one complicated good question. So this is listening and reading in one subject. Remember, all the maps I've shown you are just one subject maps. And I'll show you, we'll move to a multi-subject in just a minute. But this is just one subject. Now, your question is, well, what happens over time? There are two things that could happen over time. First of all, the person has a life, right? They're not in cotton on the shelf, and their brain might change just due to normal life, and so when you bring them back, their brain might be different. A second thing that's actually a worse problem is you only hear a new story once, and after that, it's not a new story, right? When you watch the television show the first time, it's pretty cool, and after that, it's just a rerun. And that actually, level of adaptation, that is really, really pronounced in these stories because it's actually more of a problem with, you get more of an adaptation component in these stories than you do with normal stimuli, because in a normal psychology experiment, nobody cares even the first time, right? So the subjects are always bored. Here, the first time, the subjects aren't bored at all, But by the, if you try to collect a bunch of repeats for your validation data, they get bored very, very quickly. And so one of the things we're trying to do now is to try to see if we can model the adaptation, how separable is the adaptation component from the semantic maps in these validation data over repeats, and we don't really have an answer to that yet. That's a really good question. So in this case, we're listening and reading with the same story, so some of the difference here might be due to the fact that the person's heard the story twice. But remember, because we have a common semantic space, remember, this isn't like a normal psychology experiment where the variables we manipulate in the experiment are the variables we're regressing. Here we could have two completely different stories and map them both into the same semantic space and adjust the semantic weights for the bias in the intrinsic stories because they're going to cover the semantic space differently, and then compare the two results on these semantic maps using actually two different stories. That would be a perfectly reasonable thing to do here. Do you have another question? No? You're suspicious. Good, I like that. I am the most skeptical person on the planet. You should be skeptical. But if you have a question, you can just ask me anytime. Or you can ask me now. Okay, either way. All right. So, yeah. I always say if science was easy, everyone would do it. Okay, so this attention thing is just something to keep in mind, right? This is not a learning attention effect. This is a task-based attention effect. And these attention effects are huge, and they actually, all effects of attention and task become increasingly influential as you move from the posterior pole up to prefrontal cortex. And that's just something you need to deal with. All right. So now I would like to deal with this multi-subject issue. All the maps I showed you were for one subject. It happens to be the person in our lab right now who has the best signal to noise. So if you scan everybody in your lab all the time like we do, you know who has good signal to noise and who doesn't. It's not a personal thing, it's just business, right? The main limitation on signal to noise in MRI, barring movement and things like that, which really kill your signal, are how big the person's head is relative to the helmet, right? So the MRI coils that Siemens provides us are made to fit 90% of all humans. Some humans have particularly large heads, so those coils have to be pretty large. And that means that the coils are actually pretty far away from the head for most subjects. So what you want is a subject whose head is perfectly coil-shaped, fits exactly inside the coil, and their skull is infinitely thin like Humpty Dumpty eggshell. That would be the ideal subject. So we used to have our lab tech in the lab. Brittany had a great brain, and we used her for years and years, and she would always be the first subject we would use because she had great signal to noise. She is now left, so now our best subject is Mark, my postdoc Mark Lesquois, and he's always the first subject we use. And it's our kind of a go-to person. We always use a person with the first biggest signal to noise, but you have to have multiple subjects. So here we have multiple subjects. So now you can look at these maps. These are all done in individual subjects, right? Every single thing in this experiment is done individual subjects first and then group the data afterwards, as opposed to the psychology way of doing things, which is group the data first and then see if there's individual subjects effects, which is, from any philosophy of science, exactly the backwards way to do it. You do the individual data points first and then you group. So here we have a bunch of individual brains, and now we can just take a vote. Who thinks these semantic maps are similar? Who thinks they're different? And the rest of you are asleep. Okay, fine. So, or surfing the web. So it's about 50-50, and I think that's legitimate, right? You can look, if you look at these, they're on a course level the same, right? Everybody's got a red patch in TPJ, both right and left TPJ. Everybody's gotten the same thing. Everybody's got this green penumbra around TPJ, although in some people it's almost green-ish, right? It's just barely there, but they've all kind of got this green penumbra north of TPJ. And if you look through the structure, they're reasonably consistent at a course level. But we need some objective method for aggregating these data together. And it's kind of unclear how to do that. How should we aggregate data together? So in science, we have, in MRI, this is a classic problem, aggregating data. People don't usually look at their data before they aggregate it, but they all aggregate it. So you have to, so how do they aggregate data? Well, there are two methods people use. The first is the method probably a lot of you have used at some point if you've done MRI, which is cross-subject volumetric alignment to some standard coordinate space. The standard coordinate space people usually use now is the Montreal Neurological Institute coordinate space, which is essentially the average brain from 1,000 subjects. And that's shown in the colored outline here. And now you take your individual brain and you can perform linear affine transformations to align it to this canonical brain. Or if you're really aggressive, you can do nonlinear transformations to try to warp it. That's when things get kind of spooky, though, because you know that's a completely open-ended problem that has no objective solution. So this works. You can align a brain to M and I coordinates, but it's got some problems. First of all, the M and I brain is an average of 1,000 brains, so it doesn't actually have all of the microstructure that real brains have because a bunch of that microstructure has been averaged out. And also, you know, it may not be that an affine transformation of your brain into M and I coordinates is actually the optimal thing to do. And there are usually a bunch of sulci and gyri in the brain that are not properly aligned because the M and I brain didn't actually have that information in it. So this is an okay thing to do, but whether it works really well is kind of an open issue. It's better than doing nothing. It's better than just averaging the brains without normalizing them, right? But the anatomists generally don't like this. They view it as a hack. And my old boss is an anatomist, David Van Essen, so he is one of the people that developed the other way to do this. The groups that use a surface-based alignment tend to come from anatomy. They're like Marty Serino, who is John Allman's student, and Serino and Fischl and all those guys using FreeSurfer. And then there's David Van Essen, who developed this software called Carrot that isn't actually made anymore because FreeSurfer works as well as anything. You can't really beat FreeSurfer. So in the surface-based alignment method, what happens is you extract the rind of the brain and you throw away all the little giblets, and now you have essentially a brain-shaped beach ball, and you plug an air hose into the beach ball and you blow it up to get rid of all the sulci and gyri, and then you apply some nonlinear transformations to make it into a sphere. And those are deterministic, so you can do that. It's a two-directional thing. So now I have a sphere. Now I can just rotate the sphere to align it to all the other spheres. The spheres are aligned according to where the major sulci are. And once I do that, I've now rotated everything together, and now I can basically just, I can look out in the sphere. Almost nobody does that. They actually did it at the beginning, but that was even more confusing than flat maps. So generally what they do now is they deflate it back into a brain-shaped beach ball, and they look at it there, okay? So this works better than volume-based alignment, and it has to. It's using more information than volume-based alignment, right? All of the sulci and gyri are being used here rather than just the major ones. But still, this is not a deterministic problem. There is no correct way to align these brains. It's an optimization problem. And so you don't really know if your alignment is correct or not. And any incorrect alignments, remember, are going to be lossy. They're going to lead to loss of information by averaging over subjects. And you don't ever want to average unless it's the right thing to do. Averaging is a two-edged sword. Optimal averaging is great because it gets rid of noise and keeps signal. Arbitrary averaging is bad because it gets rid of signal. And in MRI, there are a lot of cases where people do arbitrary averaging. A lot of cases, and they're all evil, 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 evil. This one is kind of the least evil because what the hell are you going to do better than this? This is a good idea, and you're kind of arbitrarily averaging over parts of the brain that shouldn't be aligned, that you decided should be aligned. But it's hard to think of what you would do differently. There are other cases in MRI where people average where I would argue they should not do it. And the classic case is when people compute statistical parametric maps using SPM or FSL or AFNI. Statistical parametric maps are one of the oldest methods of analyzing MRI data, but one of the first stages in SPM is to average your data. And you average it by smoothing it with a Gaussian whose half width is something arbitrary that you picked between, say, 2 millimeters and 8 millimeters, depending on, I don't know, what side of the bed you woke up on. So that is not optimal smoothing. That's arbitrary smoothing. And because it's arbitrary smoothing, you can imagine the optimal smoother might be different for different locations in the brain, right? The SPM smoother is designed not to optimize your signal to noise. It's designed to make sure that your statistics come out the right way. So I would argue that's a bad thing. MVPA with a searchlight, that's also smoothing. Because what is MVPA? Well, it's a vector of voxels in a certain region. How is that region determined? It's determined by the searchlight. How did you pick the searchlight? Usually you just pulled it out of your ass, right? 6 millimeters. Fine, let's try that. Well, that's smoothing. It's arbitrary smoothing, and it's the same all over the brain, and you just made it up, and you didn't cross-validate it or anything. Nobody ever does that with searchlight, and so that's bad. So you don't want to smooth things arbitrarily. You never want to smooth things unless you know it's the optimal smoothing. because MRI is all type 2 error. It's all signal you don't see. And if you're already in a regime where you're plagued by type 2 error, the worst thing to do is to make more type 2 error by smoothing away any of your signal. So you have to pay really close attention to signal-to-noise issues in MRI. And you know, our lab does a lot of spooky stuff, and people say, how did you do that? And really, a lot of it is just, well, we did the same thing you did, we just didn't smooth our data, right? So, or we didn't smooth it arbitrarily. Okay, so now, we worked for about 5 years on this subject alignment problem, trying to come up with a way to align data across subjects that was better than FreeSurfer. And we couldn't. And I spent time and money, but really, what do? I just stand around and talk. I don't actually do any real work. Graduate students spent their lives, their sweat and blood and, you know, potential career death, you know, periods, trying to solve this problem. And it was really hard and made us all very, very sad, because none of us could do anything better than FreeSurfer. So as a consequence, for years and years and years, my lab never published group average data, because we couldn't beat FreeSurfer, and I don't like FreeSurfer because I think it's doing something evil, and so we just presented all the individual subjects. And that was okay, because in Vision, that's what everybody does. In Vision, nobody averages data across subjects. You just present all the individual maps. Every other area of MRI, everybody averages across subjects and presents the group map. Anybody know why? Any guesses? Okay, I have an opinion. I mean, nobody knows why. You know, it's science. Who the hell knows why? But my opinion is, in Vision, you know what you're supposed to get. And you know that if you average a bunch of visual maps across individual subjects, your results will suck, and they'll be completely wrong, and so you don't do that. If you go up to prefrontal cortex, some complicated, interesting part of the brain where we have no idea what's going on, you don't know what you're supposed to get, so what the heck? You might as well average, because who the hell knows what you're supposed to get anyway? So I think if we knew what we were doing in more interesting parts of the brain than the visual system, we would not be averaging them. We would keep everything in the individual subject space. And another weird bias I have is that I think everybody should be publishing individual subject maps rather than group maps. You can publish a group map if you want after you show me the individual subjects, but I always want to see an individual subject. That's what we would require in Vision. Vision, I mean, things have relaxed somewhat for higher order Vision, but generally people want to see the individual subject maps. Okay. So we worked for a long time to try to come up with a way to deal with this problem. And it was all a waste of time, and it was very sad. And Alex Huth, the same guy that did the semantic experiment, had a brilliant insight maybe 4 years ago, and he realized that we were thinking about the problem the wrong way. He said, look, we're thinking about this this problem as a discriminative model. In statistics, a discriminative model is one where basically you have a distribution and you compute moments of the distribution, like the mean. And everybody who's doing alignment is basically taking all the individual data, and they're trying to align them to get some sort of adjusted, normalized mean, you know, space or mean brain or mean pattern. And we shouldn't be thinking about that way. We should be thinking about it like a generative model, which is a new area of statistics, which I'm sad to say did not even really exist when I was in school. In a generative statistical model, you don't take the individual data points and average them together to get the mean. Instead, you keep all the individual data points as individual data points. And what you try to do is estimate the underlying probability distribution that produced those data points as samples from the probability distribution. So it assumes a lot less about the distribution than a discriminative model does, because when you average in a normal statistical framework, you are making some assumption about the distribution of your data. In a generative model, you're not making assumptions about the distribution of the data. You're trying to estimate the distribution of your data. And so you can think of a generative model as creating the God brain that splurts out individual human brains from its entity, right? So how you do this is kind of spooky, and I am by far from an expert in generative modeling. This is something where we had to get Tom Griffiths involved, because Tom comes from the MIT Mike Jordan, Josh Tenenbaum empire, where they're very good at these discriminative models, and Alex worked with several other people, and Tom, and I had almost nothing to do with this, but I'll talk around it a little bit. So the idea is, okay, let's assume that each brain is like a pizza, where the toppings on the pizza are functional areas, right? And of course the problem is we're looking at this pizza in the dark, so we can't actually see the toppings. We just know that there are a bunch of toppings there. And what we want to do is take our functional measurements and then infer where all the toppings are on the pizza from those functional measurements. So if we assume that the pizza is completely covered with functional areas, we can actually replace each individual functional area by its centroid. And now we have what in physics is kind of this ball and springs model, which is a standard physics model that's used for a lot of different things, where each individual functional area is represented by its centroid, and there are springs connecting all of the adjacent areas, and the springs can have different parameters. They can have a length, and they can have a strength, right, a floppiness. Some springs are going to be really floppy, and some springs are going to be really tight, and you can't stretch them much, right? And those are going to be parameters we can fit. And now, what we're going to do is we're going to take one individual brain and we're going to pick an arbitrary number of areas. After all, we don't know how many toppings are on the pizza. So let's pick 50 areas, okay? So now we just put a bunch of little balls all over the brain that represent the centroids, and we connect them all together with arbitrary springs. And we do that for all the brains except one. And now we use standard, MCMC Gibbs sampling kind of procedure to basically go down the gradient, adjusting the length of the springs, the tension of the springs, and the position of the centroids so that they're consistent as possible across all of the brains, given the springs, right? All of the brains, this model is going to assume that all of the brains have exactly the same functional area and exactly the same topology relative to one another, but that their exact position and distance from one another is going to be dependent on these springs, which are all determined. They're determined at the group level, but an individual person can have different values for the actual instantiation of the springs. Okay? So the shared across subjects is going to be the ideal spring lengths, the spring constants, which are the springiness, and the functional means, meaning what you think. So what's going to happen in this procedure is you're going to lay down a bunch of springs, and then you're going to see how consistent those are with the data, and they're going to try to drive down a gradient to change the location of these centroids so that it more closely approximates the data. And then unique to each subject is going to be the exact centroid locations and the functional values of the functional data on cortex, of course, because that's all individually different. So this shows you the thing settling. Initially, the balls and springs are in an arbitrary state, and then you can see as it runs through hundreds and hundreds of these iterations, the individual maps, although they're generally similar with each other, they change and they're different for each individual subject. Okay. So the amazing thing is this works. This works at least as well as FreeSurfer and better in a lot of cases. So here you see the actual semantic map. Right, so there's one, again, we have to worry about overfitting here. So if you have 7 subjects, what you do is you fit this pragmatic model to 6 of the subjects, and now you take the 7th subject's data, okay? What are you going to do with that? Well, you throw away the functional data, you take the anatomy, and you give it to the pragmatic model, and the pragmatic model predicts where the functional areas should be across cortex, okay? And that's what's shown here. So here we've left out subject 1, subject 2, and subject 3. Here is the pragmatic solution for these subjects. Here is the actual functional map for these subjects. And remember, these functional data were not used at all in any of these 3 cases to fit these pragmatic data. The subject was completely held out. And these are the likelihood ratios over here on the right. Red means there's a very high likelihood that that area is correctly represented. Blue means there's a low likelihood. And you can see that the maps are mostly red in the locations where the pragmatic model actually fits. Excuse me, I should rephrase that. In the areas where the semantic model is actually predicting well, right? So in the motor and somatosensory strip, the semantic model performs poorly, and so pragmatic just makes that a big blah, it doesn't really care, right? There are a few little blue spots, and those turn out to be functional areas that you can see in the functional maps that are offset relative to where pragmatics thinks they should be in those subjects. So this works amazingly well. There is a paper on pragmatic, if you are curious. It's on arXiv. It's the mathematical algorithm that is a mathematical description of the algorithm. There is no data paper on pragmatic other than our Nature paper from April, but there are other papers coming out. The software is also not really available yet, and I wouldn't want to actually give it to a human because I think at this point it demands a lot of intervention. But we're trying as fast as we can to make this something that everybody can use. So here's the pragmatic solution for this semantic data. There are about something on the order of 70 to 100 semantically selective areas in each hemisphere. You can see the number of areas by basically just varying the number of areas in your pragmatic solution as a hyperparameter and then picking the peak of that hyperparameter or where it saturates. So there's about 70 semantic areas in each hemisphere. If you care, you can actually look to see what these different semantic areas are selective for, and you can do that by basically just taking all of the voxels in a semantically selective area like LPC11 and then looking at its selectivity across different semantic concepts, but that's nothing anybody wants to do here. It's nice though, if you look at, TPJ, this is TPJ, and this is TPJ in here as one of these things? No, I don't have it. Anyway, in each of these areas where normally people think of these as being sort of a homogeneous functional area, you can see that there's actually multiple semantically selective functional areas that are, again, present in every subject, because that's the whole point of this model. It tells you that the areas are present in every subject. So that, I think, is a pretty remarkable achievement of this model that you wouldn't get from another framework. And here we've never averaged the data across subjects. So this is consistent with my bias against averaging data. So I don't know, we have this online brain viewer. I'm just taking a poll. How many people have played with the online brain viewer from our paper in April? Only that many people? You are missing the most fun you will have that's legal. It's really awesomely cool. I'll show it after this. There's an online brain viewer for this experiment that uses our 3D graphics software called PyCortex that allows you to make these three-dimensional maps and then inflate them and deflate them in real time, and you can click on the maps and show where semantic selectivity is. I'll show you that in a second. We're also working on a replication study that will allow you basically to, since we have this pragmatic model, now you can take whole new words, like the word doofus. Okay, the word doofus was not in any of the stories that I recall. So now you could say, well, where would doofus be represented in the brain? And you can basically give this program, this replication program, the word doofus, and it will say, well, we predict doofus would be represented in all these locations in the brain. So that's coming out too. That'll also be an online tool you can use. And then I would just like to say that this, I only told you about semantics here, but you can use this method to do any kind of experiment. So one of the directions the lab's going is trying to get a small number, or ideally a lot of subjects with many hours of different tasks. I'm working on this, trying to get money to do this. I know Bertrand Thirion in France is working on this. I think there's somebody at Carnegie Mellon who's trying to work on this same problem. There's a lot of labs now that are trying to get really long data sets for individual subjects. And so this is a data set where we have 2 hours of narrative stories, a couple hours of short films, and several hours of decision tasks. And you can actually fit all of these different tasks together simultaneously using feature spaces, multiple feature spaces that are appropriate for each task, and you can make very rich maps, again, of the voxel selectivity under these different tasks. So I told you this was cool, and that's about it. Since I'm over time, I'm going to stop. But I did want to show you this because you should really check it out. It's always tricky to do this online. Okay, so, okay, here we go. Good. And then F11. Okay, so we got a brain here. This is online. It's at gownlab.org. You can just find it under Brain Viewer. So there's a brain. Fine. You can blow it up. You can move it around. Let's see. Okay, so if we look back here in parietal cortex, okay, some of you, God, is this just me, or is it's really warped? Is it warped for everybody in some weird way? No? God, it's so foreshortened. Okay, so there's this big purple patch here. Let's click on that. And now you can see, all right, just do it as homework. Screw it. It's gownlab.org. Go there and you can play with it. Anyway, that's it. Thanks.
Jack's next topic is controversies in functional imaging, which is interesting because I thought that was the topic of the first talk. Because as you obviously know, what we taught you for the last 8 days is wrong.
I didn't say it's all wrong. I've gotten over that.
Yeah, well, the need to say it. Nevertheless, the message was there, Jack. So,
what?
Anyway.
I'm not trying. I'm trying to be a positive person. I didn't insult anybody by name. I thought I was being particularly good.
All right, we're ready. We're ready when you are, of course.
Hang on. I have to export this thing to you, Mark. Oh, I did that. It's 11. Okay, wait. Okay, since Mark accused me of saying everybody was wrong, I should mention on the semantic selectivity paper, the results are really interesting. The results do not contradict any other study. I mean, they may contradict specific studies, but no generally agreed upon result, right? So if you look at where people think number-related areas are, in our semantic maps, that's where the number-related areas are. If you look at TPJ, all of the concepts in TPJ are social-related concepts. That's generally consistent for all of the semantic selectivity. So if there is some functional assignment of a region, the semantic selectivity of that region for narrative semantics will be directly related to the functional assignment of that region in general, if it's responding at all to narrative semantics. So I don't think what we showed was that other people were wrong. I think it's not a matter of wrong versus a right. It's a matter of how much data you get out of your experiment. And, MRI is pretty expensive. You guys may not notice it yet because you're graduate students, but your advisors definitely notice it. I don't know, MRI pretty much, just to pay for your magnet, is going to be about $300 an hour. If you manage to get a really rich donor who, why don't you just turn up the volume, Mark? Oh, all right. This just feels very unnatural. I do not like hearing myself. So if you have a rich donor who pays for your magnet, then you can, like some of the nice, cushy Ivy League institutions have, then you can scan for free and you don't notice it. But for most people, if your magnet's at a hospital or your magnet's at a state school like ours is, then you're going to have to pay for that time. And at $300 an hour, that's a lot of time. Plus, you have to pay the subject on top of that. At our scanner, it's actually much more expensive than $300 an hour because we have staff that we pay. So this adds up really, really quickly. So in something that is this costly, my view is that it is incumbent on you, the user, certainly if you're paying for it yourself, to be as efficient as possible. So when we do an experiment, I think of experimental design in a weird way that nobody else does. I think about bits per dollar. How many bits of information am I getting per dollar I spend? And by bits, I mean I want to calculate the mutual information between the things I manipulated in my experiment and the things I measured. So, right, remember mutual information is the difference in entropy between one end of your communication channel and the other. So in this context, the communication channel is the MRI machine. So I've got some entropy in my experiment. If I have two conditions, then I have essentially 1 bit of entropy. So I have a very low possible information I can recover from each voxel. I can only recover 1 bit, right? If I use a natural story, then I have a lot of entropy in my input end, okay? So now I send this information through my channel, which is my MRI machine, and I measure some entropy at the end. Now, MRI, you know, I'll make this really quick. Your MRI machine has 20-bit DACs. The files that Siemens uses to store the data are not 20-bit files, they're 16-bit files. The NIFTY format is not 16-bit file format, it's 12 bits. Siemens decided many years ago that they didn't really need 12 bits because you didn't deserve them, so they were going to save 4 bits for themselves, so they only give you 8 bits. Now Siemens gave you the best 8 bits, they have class, but they're still only 8 bits. And then on top of that, actually not all of that is real data. Basically you get about 5 bits. Each TR you get about 5 bits per voxel. So you have a bunch of entropy in your input, you have a bunch of entropy on your output. You can calculate that, you can integrate that over an hour, and you can calculate how many dollars you're paying for each bit of information. And I would argue that you need to optimize that because otherwise you're wasting money. And if you're wasting money, you're probably wasting time too, and nobody wants to waste money or time. So even though this approach seems like it's more work, I would argue it's actually less work than the normal way of doing things. All right. We're going to just get through as much of this as we can. So I wanted to rant for about 10 minutes, which might be almost all the time I have, on the replication crisis we have in neuroscience. Now, you are probably all familiar with this if you're on Twitter or plugged into any crappy Murdoch newspaper. That there was a recent study published by Tom Nichols' group that basically said that all MRI studies published in the last 20 years were wrong. That was essentially the press summary said that 40,000 published MRI studies were to be questioned because of type 1 error inflation, dramatic type 1 error inflation. Now, unfortunately, Tom and his friends did a really good job of getting attention from the press, not really the kind of attention we want, the kind of negative attention saying MRI is all crap, but they had actually overstated the case. They had done their simulations to show that there's type 1 error inflation in one particular form of cluster analysis of SPM data, which probably on the more recent correction that Tom published last week is probably only affects at most 3,500 studies. But still, 3,500 is a lot, right? And these are dramatic inflations of type 1 error, right? Where the false positive rate, instead of being 5% like you would like, are going to be more like 70%. And their argument is this is bad. You know, we don't want to have a bunch of false positives because that means we're going to have a replication problem. And you guys have all seen variants of this from, you know, the dead salmon experiment and so on. All these experiments claiming that there's something fundamentally wrong with MRI. And it's always ambiguous whether something's fundamentally wrong with MRI or something's fundamentally wrong with the way we do MRI. And I would argue it's the latter. It's not the MRI that's the problem, it's what we're doing with the MRI is the problem. And actually, this isn't a problem that just affects MRI, this affects all of biomedicine. So the person who's most famous for pointing this out is John Ioannidis, who's now at Stanford. I think he did a lot of his early stuff at Tufts. But basically, he's been pointing out for years and years and years that type 1, that due to a combination of problems, statistical significance is, in practice, inflating our type 1 error rate far beyond what we would expect. Remember, type 1 error is reporting that there's a significant result when there is not a significant result. And Ioannidis and other people have pointed out that actually the type 1 error rate, even though you think it's 5%, is actually much higher in all of the reported literature. And the reason has to do with the perverse incentives of science. Basically, you can publish a positive result, you can't publish a null result. People have to publish or they perish. So therefore, people do everything they can to publish a positive result. Well, what can you do to publish a positive result? Well, there's a million things you can do. You can run your study not double-blinded. You can p-hack and try a bunch of different statistical tests until you get one that works. There's a million things you can do that will give you a significant result, but actually give you a high type 1 error rate. And every possible way you can scam significance values, you can bet there are multiple people out there using that particular scam, either consciously or unconsciously, to inflate their significance values, because that's what science is rewarded for. So this is a serious problem, and it's a serious problem in all of biomedicine and psychology. Anybody, basically, any field which is relying on significance rather than predictive models has this problem. I should note that this goes way back. People have known there was a problem in this, of this form for a very, very long time. Here's a 1997 paper about psychology. But actually, the first people who noticed that there was a problem with statistical significance were Neyman and Pearson, who came along soon after Sir Ronald Fisher proposed statistical significance as a test. And people were interpreting significance values as continuous variables, as essentially giving you a confidence that the result was real, which is not what significance tells you. And Neyman and Pearson were the ones that came along and corrected it by introducing the decision criteria. And our use of statistical significance today is sort of the Frankenstein stepchild of a combination of Ronald Fisher and Neyman Pearson. We don't actually follow either one. We follow some aggregate average of both of them, which is not a good thing to do. So this is an old problem that we've known about for a very long time. There are a lot of different aspects of this problem. I actually just put this paper in here. This is there are two people who have written a lot on this. One is Ioannidis, who's at Stanford. One is Gelman, who's at Princeton, I think. Mostly not about MRI, but more generally about psychology. Gelman has done a lot of psychology stuff, and Ioannidis has done a lot of biomedicine stuff. And I actually just put this paper up here because I like the title of it, because it's what I say a lot. Statistical significance is not significant. And the reason people think statistical significance is significant is because it says significance right in the title. It's the same reason people think that functional connectivity is connectivity, because it says connectivity right in the title, so it must be connectivity. It's not. It's not connectivity, and it's not significance. And more importantly, statistical significance is not importance and has nothing to do with importance or effect size, nothing whatsoever. A significant result has no relationship to or impact on how much of the variance your model will predict. There are a bunch of people who have fought back against, this is from Harold Pashler, who's done a lot of work at UCSD on this issue as well. There have been a lot of people fighting back with various arguments about this replication crisis we have and this inflation of type 1 error. The arguments have been of multiple forms. For example, well, it's really not that bad a problem, or really if you have a really strong theory, then it's not that bad a problem, or it doesn't matter, we don't replicate anything, because after all, if you replicate with two different methods and they kind of give you the same result, when you squint, that's good enough. All of these arguments are made. They're all, in my opinion, invalid. This is a really serious problem. The problem is, if you can't trust what other people publish to be real, how are we ever going to make progress in science? We can't replicate anything. We can't replicate everything. It's impossible to replicate everything. We would have to use all the resources of the planet to replicate everything. That's an insane thing to do. What we need to do is change our systems so that we can trust without needing to replicate every single thing. More studies of the same thing. Another aspect of this that's been known for a long time is the file drawer phenomenon, right? So remember, the file drawer phenomenon is you do an experiment and it's not significant, so you put it in the file drawer, you don't publish it. You do an experiment, it is significant, then you publish it. So if all that happened was that there were 20 times more experiments than published experiments, then we could infer that almost every result that was published could likely have been due to chance. Now, of course, things aren't probably that bad. It's probably more like there's 3 times as many experiments as there are published experiments. But that still means your type 1 error rate is a lot higher than you think. So there have been since everybody has acknowledged we have a problem, there have been a lot of proposals for how to deal with this problem. Some of them I think are smart and good, and some of them are not. The first is that all data should be open. We should just open up all the data to everybody immediately upon publication, because then everybody can just check everybody else's data and make sure that it's right, make sure they didn't make any mistakes. And in an ideal world where we all had infinite time, that would be a great thing to do. And I actually do believe in open data because the taxpayers paid for all of these data, and therefore they should be available for others to use. But just opening data won't solve the replication problem on its own, because I have had open data out on the web for a long time, and I can tell you that very few people actually use those open data or check them. So just having the data available won't cause people to check the data unless there's already suspicion that there's something wrong with the data. There are other problems with opening data that mainly have to do with attribution. So for example, in this semantics experiment that I told you about earlier, we're probably going to publish, just based on the papers we already have mapped out, 5 or 6 papers on that same data set. So if the requirement for us was that all of the data be opened up from the very first paper that we publish, that would mean that all of the other work that could be done by my students and my postdocs who actually suffered for that data would be done by somebody else, and I consider that to be unfair to my students and postdocs. So I think the open data initiative is a great thing to do, and I think once you finish publishing all the primary papers, your data should be opened. That's the policy we follow in our lab, and I think that's the policy that all labs should follow. All of our data is on a resource called crcns.org, which is collaborative research in computational neuroscience. It's run by the NSF. And our neurophysiology data is put up there, and our MRI data is put up there. But there are other avenues for pushing your data. You can put your data on NeuroVault, for example, and it'll just be there for anybody to use. So I would argue that the only thing that really matters in all of this is not statistical significance, but effect size. And you can calculate effect size using classical statistical metrics, which involve a lot of assumptions, or you can calculate what you really wanted, rather than effect size, which is prediction accuracy. The only thing that really matters in science is prediction accuracy, because of course the whole point of science is to build a model that predicts. The only reason the government is paying us any money is to build models that predict. When they're not paying us to enjoy ourselves, the government doesn't care whether we enjoy ourselves. And science certainly doesn't care. So really the reason we have jobs is to build models that predict. And if we're not building models that predict, then we're not doing our jobs and we shouldn't be getting money. And so we have to be reporting predictions and effect size. And that means that since the only way I would argue to estimate prediction accuracy or effect size correctly is to collect separate estimation and validation data sets, or as they're sometimes known, separate fit and test data sets, then I would argue that every single study needs to be collecting 2 data sets, a fit set and a test set. If you don't do that, you can't have any guarantee at all that you're not just overfitting the heck out of your data. So in my perfect world, first of all, every single journal would require reports of effect size. The number of journals that require reports of effect size today is, as far as I know, 0. So we have some ways to go to do that. And secondly, every study would report effect size the right way by collecting 2 data sets. Although that might not always be possible, it should usually be possible. But I'm not as adamant about that as I am about just journals requiring it. I think journals should always require effect size, and if they did, that most of this replication crisis would go away. All right, so talk a little bit about measurement technology. In any area of science, there's kind of 2 things that limit you. One is theory and one is measurement. And of course, in every area of science, both of those things limit you all the time. But in any specific area of science, one of those things is limiting you at any specific point in time. And in MRI, many people think that the main problem we have with MRI and understanding MRI data is theory. We just don't have good enough psychological theories. I would argue that's not the problem. The problem is our measurement technology. That MRI is an amazing device. It works way better than anything else we have for non-invasive measurement of human brain activity, but it's fundamentally just collecting a pale shadow of all the data we need. And if we really want to have any hope of building really good predictive models, we need to improve MRI. And since we're not going to be able to improve the time resolution of MRI as long as we're relying on hemodynamics, and it's unlikely we'll be able to get that past that soon, although Mark may have some magic up his sleeve he hasn't told me about. The one thing we probably can do is increase the spatial resolution. So, you know, what we really like to do is get to this level. Here up on the top is a voltage-sensitive dye and a calcium imaging map from Clay Reed's lab showing that individual neurons are orientation tuned according to the orientation map you get from voltage-sensitive dyes. So this suggests that if our MRI machine could recover something at the level of a cortical column, a sort of a voltage-sensitive dye level measurement, that we could recover a lot of this local information that's buried in these functional maps that we can't currently see, because current MRI at the level of 2 or 3 millimeters is measuring essentially a whole hypercolumn, a bunch of columns together, and all the individual attributes of those columns are all averaged together and averaged away. So that would be what we would like to do. Both fMRI and electrocorticography are kind of limited right now in that they can only record hypercolumns. MRI, there are a variety of methods we might use to try to get down in spatial resolution, and there are several groups working to create new electrocorticography grids for doing this in humans to get down to the column level. Right, this is my hypercolumn slide. So there have been several attempts, actually, almost since the dawn of MRI, to try to do columnar imaging and to try to recover sort of the functional properties of columns. The first one that I know of was by Ravi Menon, who tried to look for ocular dominance columns in primary visual cortex. And there is a reason for looking at ocular dominance columns, because ocular dominance is organized at a larger scale than is orientation. So ocular dominance columns are closer to the scale of a hypercolumn, and so you might guess that you could recover ocular dominance columns. If you read this paper, which is a great attempt to recover this information, you may well decide that they probably weren't successful in doing it. But people have continued to try to do this, so more frequently, here's a paper from the Minnesota group, Essa Yakub and Camille Urabil and those guys, where they also tried to recover ocular dominance columns. And this paper suggests that now in 2007, we can probably maybe get ocular dominance columns. But of course, ocular dominance columns are not the level you want because they're organized at a scale larger than regular cortical columns. You need to go down to the scale of orientation columns to really have something that you feel confident you could use to recover columnar architecture across the whole brain. So there have been attempts to recover orientation columns. In fact, the Minnesota group published a nice paper a few years ago trying to recover orientation columns. So this is their ocular dominance map on the left. This is their orientation map on the right on a flattened V1. And they argued in this paper, oh, look, we can get orientation columns, we can get ocular dominance columns, and we can get orientation columns. MRI's awesome, we rule. And that's great, you know, but if you really had orientation columns, you would assume that when you measure them across 2 different days, the pattern would be the same. And they very nicely provided the information across days. Here on the left is day 1, here on the right is day 2. Oops, wait. Is it day 1 and day 1? Oh, sorry, here it's the top and bottom, I guess. It's day 1 and day 2. So here are their 2 orientation column maps. They're supposed to be aligned, and so they should look the same. And as you can tell, these don't look the same, right? These are not the same maps, which probably means that just, you know, nice try, but no orientation columns. And I don't know of any study since then that has done any better than this. So we don't have any MRI signals yet that are at a spatial resolution that we can pull out columns. So there are several attempts to do this. I'm involved with one of them, with this guy, Dave Feinberg, who's running kind of a consortium grant with the people at Martino Center are building the coils. I'm involved in evaluating stuff since I don't actually do any coil building. There are several other groups involved. And the idea is to basically, it's really the dumbest idea in the world. Let's make tinier coils. After all, if we have just one big coil in the brain, we get low resolution MRI. If we go to 12 channels or 32 channels, we get higher resolution MRI. What happens if we go to, say, 256 channels? Well, we might have a problem with depth, but if we really care about cortical imaging, we should be able to maybe record columns at a really high spatial resolution. So this is what they've been working on. This just shows you, I can't remember, to be honest, if this is real data or simulated data. So I'm going to be agnostic about whether it's real or simulated in this talk. So here's a 12-channel coil. Here's a 32-channel coil, which is your standard high-end Siemens coil. And here is, what does that say, a 96-channel coil, right? And so you can see the signal-to-noise here should be higher on the outer sort of rind of cortex. And then by the time you get down to the middle of the brain, the signal-to-noise won't be any better or will even be worse because, of course, the sensitivity of a coil is proportional to its diameter, right? The depth sensitivity. Now, if this was real data, it would have been built by the little coil building gnomes in the Martino Center, and maybe it would have had an active shimming, mechanism in it, and so on, to be a really high-resolution coil system. But there are other things you can do that might even be better. So you remember I mentioned earlier that one of the biggest, I'm going to be done in like 2 minutes, Mark. One of the biggest problems in MRI is the distance of the brain from the coil, right? And if you use any standard coil that is made for, a large number of humans, it's going to be much bigger than most people's heads. And one easy way to increase signal to noise is not to make tinier and tinier coils, but to make coils that go right on the person's head. And you could imagine a system where you would actually customize the coils for each individual subject. And that's another direction of this grant that's being done with Mickey Lustig and our lab at Berkeley. Mickey started making these flexible coils. He actually made them because he wanted to sew them into baby blankets to do neonatal imaging. So now instead of the babies, the problem is they're changing their size pretty radically over a very short span of time, and no hospital can afford an array of baby coils that are optimized for all sizes of babies, right? So the idea is, well, what if we make flexible coils in a blanket and just wrap it right over the baby, and then the coils will always be as next to the baby as possible, and you can solve the problem that way. Now, this has issues because the coils are now no longer in a known geometry, so you have to use tricky statistical methods for deconvolving the data and reconstructing the images, but that's actually what Mickey specializes in, so he's not afraid of that. So once you have these flexible coils, which are actually just made with a silk screening process, you can do a dumb experiment like just tape them on the back of somebody's head. So you tape them on their head, you stick them in the MRI machine, and you get an anatomical image, and you can see this anatomical image actually looks really, really good. Taping 4 big coils on the back of somebody's head basically gives you an anatomical image of the back of the head as good as Siemens' $200,000 coil, okay? So this suggests that this might be a really good avenue to pursue. And we're pursuing this by combining it with a little project that we've been working on for a while that we call the head case. Basically what we do is we take a laser scan of your head, and we take a laser scan of the 32-channel coil, and then we use a 3D printing process, or now we actually mill these things, and we mill a helmet that that is just for you, and it fits directly around your head, and it fits inside of the coil. So now you can't move because all of the space between your head and the coil is filled with plastic. So this prevents motion. It prevents motion even in our subjects who have been in the magnet for 200 hours. They still move. They cough, they breathe, they swallow. So this prevents a lot of the motion, and it makes sure that if you do a multi-session MRI experiment where the person comes out of the magnet and goes back in, now their head is always in the isocenter of the coils. So you don't end up with the hot spots of the coils shifting across sessions, which is one of the main problems you have with cross-session alignment. So this thing works great. I should just mention that the guy who's been working on this project for the last 2 years is starting a company called CaseForge to build these things. And the way this will work is, say UCLA wants to have these flexible coils, these head cases. So James Gao will send you a cell phone with a laser scanner on it. You take the person's head, you put a swimming cap on them, you take the laser scanner, you scan their head. The software on the phone sends it back to Berkeley. There's an automated milling machine that automatically takes the scan, adjusts it all, sticks in the middle of the coil, figures out the CAD/CAM model for the milling machine, and then mills it out of foam, and then it goes in the mail the next day and gets sent to you by next day air. So you have it tomorrow. So that should work, and basically all of the pieces of this are together, and we're beta testing this now. But that has nothing to do with the interesting part, which is these flexible coils. In the end, what we want is to be able to take these customized individual head cases and combine them with customized individual silkscreen printed coils to optimize the signal to noise for any individual subject's brain and for any individual experiment. So that's about it. Because I'm over time, that's all I'm going to talk about. Thanks for your time. Lunch. Lunch? Questions? Lunch?
Questions?
Coffee? Coffee.
Questions? Today is Pamela Douglas. Pamela is one of the many people who's been helping to organize this course altogether, and actually for several years. She's been working at UCLA since her graduate school, and is now continuing very fine work in machine learning. She's an engineer by training, and has made really interesting contributions to the field of machine learning and MRI. So this is great. Pamela Douglas.
Yay. We might need a bit of help here getting the slides.
Oh, okay, great. Here on the one that's making the projector. Okay. Let's see. There it is.
Okay.
Okay.
Yes, the slide should be posted shortly. Okay, great. Thank you all so much for having me. This is really fun. I think I've met most of you, so but I'll just introduce myself again. I'm Pamela, in case you haven't met me, Douglas. And you've heard a lot of lectures on machine learning so far, so by the end of this course, you all are going to be experts in machine learning algorithms and everything decoding related to fMRI and neuroimaging. So I'm just going to kind of expand upon a lot of the things that Jesse Rissman talked about and kind of build on that and recapitulate a lot of the things that he said, because sometimes it's nice to hear things a couple of times from a couple of different perspectives. So first we'll start off just talking a little bit about what do weight vectors mean, and I'm specifically going to talk how they relate to SVM, because support vector machines are kind of the most popular and used most commonly in fMRI decoding. And then I'm going to talk about something that's slightly controversial. Feature selection, is it important? Again, Jesse did introduce this topic, so I'm not going to go through the basics again, but I'll talk about a few of the issues that are kind of interesting related to feature selection. And then I'm going to talk a little bit about what can you do beyond support vector machine. And then lastly, I'll give this plug for this amazing toolbox that comes out of New Zealand, some of my colleagues in New Zealand, and they're a lot of fun. So. Okay, so weight vectors and SVM. So first, you've heard a lot about, at this point, voxel-based encoding models and also decoding models. And so at this point, you're all well versed in knowing that encoding models are kind of asking how activity varies when there are changes in the outside world, and conversely, decoding are asking more, what can we learn about the outside world by observing activity? These are kind of the conventional ways of thinking about encoding and decoding. And they're both essentially ways of mapping data to either the experimental manipulation or the stimulus. So early on, one of the motivations for running decoding models was this idea that we might be able to learn more about patchy functional segregation in the brain. So, of course, decoding is inherently multivariate, so the idea was that we might be able to analyze these patterns and be able to learn more knowledge. So Jesse's gone through this quite a bit, but I'll just mention that again. So again, why should we use decoding? The idea is that traditional mass univariate modeling might be limited, and that information might exist in distributed patterns. And MVPA and also just decoding methods may therefore be useful for examining this distributed coding. And so this is from Etzel, 2013, and this is just showing the number of publications that are related to decoding that have increased over the years. And this is kind of similar to the slide that Russ Poldrack showed, showing that we've just really expanded the number of publications related to fMRI over the past few decades. But we've also been publishing a lot in the field. at this convergence of machine learning and neuroimaging. So that's kind of an interesting trend. So there's obviously a lot more Haxby references, Krieger-Scores references that have been taking place over the past few years. So what can we do with decoding? Most people want to do stimulus decoding by response pattern classification, which was that first mapping that I showed you. Which stimulus items elicit patterns that are distinguishable? That's perhaps a question that you're trying to ask, because at the level of voxels, the resolution of a voxel, can we actually distinguish between different patterns? If you can't, then maybe your decoding is kind of a trivial thing. Even if you're trying to predict brain patterns, which I'll mention later, if they're so similar, it might actually be fairly trivial. You could also ask, which items does a region treat as identical and which does it treat as distinct? So that's kind of another way of reframing that same question. But more traditionally, kind of prior to fMRI, two of the things that were commonly asked in the machine learning community with, say, EEG data or maybe just structural MRI data were, can we predict group membership? So can we do computer-aided diagnostics, for example? Or can we do brain-machine interfaces? Can we use EEG, say, to control robotic arms? I saw a really interesting presentation once from a group in Japan that had built an entire apartment, actually, for people that were locked in. And so they had people using EEG to control everything from moving around a wheelchair to changing the television channels to turning the lights on and off. It was actually completely amazing. But what's also amazing is that the volunteers, who were people like us volunteering in this experiment, all threw up their hands and just couldn't take it after, a couple of hours, sometimes a day, just because it's really frustrating if it's not working and you're really trying to move the wheelchair and just so it's really challenging, brain-machine interfaces, brain-computer interfaces, in part because the brain is, of course, non-stationary. So a couple of years ago, Klaus Robert Müller came here and spoke about brain-computer interfaces, and that was a really fascinating talk on how we can deal with the fact that the brain is always remapping itself. You might recall, if you've read some of these early papers on brain-computer interfaces, even when primates are used, usually you have to retrain the neural network, or if you're using a neural network or any type of classifier each day, because overnight, even in the motor cortex, there's a lot of remapping and a lot of non-stationarities that happen. So by now you all know this. What is machine learning? It's a branch of artificial intelligence that takes in data and tries to learn and make predictions. But there's really two main classes. There's supervised, where we know the label on some training data, and then we apply that to some test data. But there's also unsupervised. This seems to receive, I think, less attention in our field, but it is also really fascinating. I've done some work with Ariana Anderson here at UCLA recently, using unsupervised non-negative matrix factorization to try and understand the dimensions of ADHD, because it's a very difficult disease to try and categorize into its different presentations, since hyperactivity and impulsivity and inattention seem to kind of overlap and span, and it's hard to label a specific patient as ADHD with an inattentive subtype, or now they're called presentations. So that is kind of an interesting aspect of unsupervised machine learning. So what do machine learning classifiers operate on? They operate on features, which are descriptive variable categories. Most of the time in this course, we've been talking about using voxels, say, as our features. But you can also have things like gender, male, female, IQ. You can have all kinds of different categorical variables. You can have missing data. So it's not just numerical values of voxels. There's a number of different attributes or features. These two words are used fairly interchangeably in the machine learning literature to try and make predictions. So here's a few examples of some common features that are used in decoding. Voxels, as we've talked about. Searchlights, which are really nice in the sense that they try to reduce local redundancy, which we know to be the case in a lot of fMRI measurements. There's also classification based on functional connectivity matrices. You can also use graph theoretic metrics as features if you like. I've seen some papers using effective connectivity, DCM parameters to make predictions and diagnostic categorizations, and also independent components. So just to summarize the analysis flow, we have an input space, say some voxels. We map that to a feature space. We then perform a classification. And then we have this output, which is a % accuracy of how well we were able to make predictions on the test set, as well as these feature weights. And these feature weights can be mapped back to the brain, but that's what I'm going to talk about is, should we really be doing that? Why or why not? So one thing to think about, with each machine learning algorithm, there's an inductive bias. So machine learning is kind of an ill-posed problem. There's no one solution. So in order to constrain the problem, we have to make some assumptions, and this comes in the form of the inductive bias. So if you have this data, part of the inductive bias could be, do we want to try and separate it with a line? Do we want to use a curve? Would we like to use a higher order polynomial? How do we choose between these different inductive biases? So some inductive biases are parametric. So if you're doing Bayesian classification, you're assuming essentially that the distributions adhere to a Gaussian distribution, for example. But not all inductive biases make assumptions about the underlying distributions. So some of the assumptions could be just based on what type of discriminant or boundary is used to separate the different classes. So those are non-parametric. So most of what I've seen people apply in the fMRI world is non-parametric because most people are using linear discriminant or support vector machine. So by far the most common that I've seen applied is the linear support vector machine. This is useful because it is very computationally effective when you're dealing with high dimensional data, like with fMRI, in part because, as I said, it doesn't depend on characterizing the entire distributions, but rather just looking at the points which are close to the decision boundary, as we'll see. So with SVM, or any linear classifier, you first define the model. So in this case, X is the input, theta is the model parameters. And here the model parameters, W is the weight vector, and W naught is the threshold. So this model here is our inductive bias. The form of our model is the inductive bias that we're taking. From there, you define a loss function. Often this is just the difference between the desired output and the model approximation. And then in order to estimate these parameters, we have some sort of an optimization procedure. So this often happens where you initialize some random weights. Maybe they're not random, but sometimes they're random, the initialization. And then you perform some sort of optimization procedure to iteratively change the weights as well as the thresholds. And so an example of this would be gradient descent, where you take the gradient of the loss function or the cost function with respect to each parameter, and then you update or you change those parameters within a loop. Usually, it doesn't have to be a loop, but most often. In this case, if you're using gradient descent, you would update the parameters against the gradient. And finally, once you have your model tuned on your training data, you then apply the tuned model to the test data. So as I said, Jesse Rissman has talked about this quite a bit, but I'm going to try to provide a geometric description of what this really means. So it's easy to first think about the Fisher's linear discriminant analysis, or any LDA kind of procedure. So it's also easy to think about it in 2 dimensions, or 3, but I like to take the simplest case because I think it's the most clear, effective way to visualize things. It's hard to imagine n-dimensional space, so we'll start with 2. So in this case, we have stimulus class 1 and stimulus class 2. Each has their own distinctive means. And we're trying to find the weight vector as well as the threshold such that when the data are projected onto that weight vector, that the data can be maximally separated. And so here's an example of a very poor choice of a decision boundary as well as a W vector. If we choose this W vector here, when the data are projected, you really can't get much class discrimination here. So this is true for SVM as well as linear discriminant analysis. The big point here is to just note that this W vector is orthogonal to the decision boundary. So what happens is these points, you can think of it as though they're being projected onto the W vector, and then you later apply this decision boundary. So here's an example of a better choice for a decision boundary. So Fisher's linear discriminant and LDA are all really nicely described in a paper from Klaus Müller's group by Lem. It's a neuroimage paper in 2011. So if you're interested in understanding the nuances of how these boundaries are chosen between the different LDA learning algorithms, then I highly recommend that paper. So with SVM, we're interested, again, with using the same model, but this time we're interested not just in separating the two classes, but finding the maximum margin hyperplane. So we're interested in separating the two classes some distance away, and we want to maximize that distance. So this is some math. If you don't like math, then don't worry about it. I heard someone once say, if you don't like it, you can just think about it as decoration. So if you're not into math, then maybe this is just decoration. But if you like it, then maybe you want to read the Vapnik and Cortez 1995 paper. There's a nice long appendix there. But as I said, we're interested in maximizing the distance between the points, and we want it to be some distance. So it's often useful to constrain the problem by setting the distance times the norm of the W vector equal to 1. And then it becomes a Lagrangian problem. So you might recall from your calculus that when you're trying to maximize or minimize a function with respect to another function, then you can use these Lagrange multipliers, and that's one way to solve this problem. But the interesting thing about this is that in this process, most of the data points don't have any influence on the selection of the decision boundary. And so most of them vanish. The ones that we're left with are the points in an optimal case, not in the case where we have the soft margin, but in the optimal case, the points that we're left with are the points that lie along the decision boundaries. And those points are called the support vectors. So when calculating the actual W vector, it's useful to average across a number of these support vector points and their parameters just for numerical stability. So for that reason, that's why it's called a support vector machine, is because it's a machine, I guess, of all of these different points that are the support vectors. So the geometry of SVM is, again, very similar to the linear discriminant analysis, the LDA approach. We have two different classes. We have our W vector, which is orthogonal to the decision boundary. We're trying to maximize that boundary. And so here are those points that I was talking about, the ones that actually make an influence, which are the ones closest to the decision boundary, and those are the support vectors. So I guess I'll repeat this just one last time, just because now I'm about to explain why it's difficult to interpret the weight vector. But again, just note that this W vector is, again, orthogonal to the decision boundary. So this is a worst-case scenario that was pointed out by Stefan Halfa in 2014. It was really also pointed out by Guyon and Iseloff in 2002, which is that interpreting these weight vectors doesn't really make sense. And so it's kind of worth spending a moment thinking about what this figure actually means. So this is, again, supposing that we have two different channels, x1 and x2. It's very clear here that x2, if you project the data onto x2, it really doesn't have any class-specific information. On the other hand, if you project the data down onto x1, it absolutely contains a lot of class-specific discriminatory information. But if you put these two in a classic LDA kind of scheme, what ends up happening is that the weight assigned to x2 is, in this particular example, is about twice that of x1. And so this is, again, kind of the worst case scenario, but it can happen that a channel or a voxel that has no information on its own is assigned a very strong weight. And part of that has to do with the covariance between the two. So if the two were orthogonal, then the covariance is 0, and this is not a problem. But it is a problem when there's shared information between the two. It can also be a problem when you have multiple voxels. One voxel that's a noise voxel can be given a very, very strong weight because it's essentially absorbing all of the noise that's shared across all of the different voxels. So this is an important point. So it's a little bit tricky, but if you do map your weight vector back onto the brain and you present it like this, even though you're not necessarily interpreting the weights, I think it can, on some level, be considered an implied interpretation because these sort of look like maybe the results you would get from a GLM analysis. But they're not. They're weights from a machine learning analysis. So what can we do with the weights to try and interpret them? Well, Stefan Haufe says that what we can do is take the weight vector and back project it into the original space. And so if you have a square matrix and a very simple problem and a linear classifier, that turns out to be the inverse transpose of the weight vector. So you can just take the weight vector, take the inverse transpose, and there you go. Then you can just show the weights if you want to. And there's nothing wrong with that, but there are some complications actually, because if you have a lot of redundancy, the weights that are assigned to the voxels that actually do contain a lot of information kind of fall off. And so with more redundancy, this becomes very problematic in how do we interpret these weights. It also becomes more complicated when you have perhaps different kernels, as Jesse talked about. You can use radial basis functions or polynomial kernels. It becomes a little bit more tricky. Or with, say, neural nets, how do you get back to that original space? How do you make sense of those features? And how do you interpret them in the context of whatever cognitive process you're trying to probe with an experimental manipulation? So, yes. So this is kind of inspired by something that Russ Poldrack wrote in 2006. He talked about the dangers of reverse inference. So one of the dangers of reverse inference is looking at functional activation maps and making inferences that those have implications for cognitive processes, as opposed to just the experimental manipulation. So there's something similar with machine learning in the sense that we don't want to take the weights, as I've said, and project those back and then make inferences based on the weight maps. So be careful with what you say with your machine learning weights. So just to summarize this, the results should always be interpreted in the context of the inductive bias that you've used for your model. Decoding may also provide insight into the multivariate nature of neural code, but I'm not exactly sure how. That's still kind of an open question for me. And although it's tempting, the weights that are assigned to features of voxels should not be interpreted directly, unless they're independent. So that's something something to consider. Okay, so now I'm going to talk about, now that we have our geometric interpretation of SVM and some classifiers, I'm going to talk about if feature selection is or is not important. So feature selection can be performed either as a separate step or alternatively as an internal step within a regularization scheme. So if it's performed as a separate step, there's a couple of different ways that this can take place. The first is maybe just a filtering approach, where you perhaps have all of your voxels and you're interested in just looking at, on the testing data, those voxels that have some certain z-score. Or alternatively, you could threshold just the ones that have more than a certain level of variance. So I think I saw this in one of the earlier papers on decoding fMRI. They just were interested in voxels that actually changed. So they had a variance threshold. So that's almost like a pre-processing step. You can also use wrapper methods. So an example of this is support vector machine recursive feature elimination. So in this case, what you do is you wrap the classification algorithm within a nested cross-validation, which I'll talk about. And you try and remove, sometimes one at a time, sometimes you randomly select groups, and you see what happens if you remove those. Does the error increase or decrease if you remove groups of features? And then there's also projection methods. There's you can use PCA or ICA to try and project clusters of features that are similar into a lower dimensional space. One that I really like recently that I'm still learning about is random projections. It turns out that this technique is not actually that random. It finds clusters that are really nicely separated. And clusters that are well separated on just a few data instances. So, for example, if you're trying to discriminate between tigers and hammers, it might be very, very different. And so you can group those and project all of those clusters down into smaller dimensions. So if you're not using feature selection as a separate step, you can also do feature selection in a way within a regularization scheme. So with regularization, you just modify your cost function. So you have the data misfit, but you also have some sort of regularization parameter that penalizes for complexity. So in this sense, the regularization is trying to move your model towards a more parsimonious model fit by penalizing the addition of extra parameters. So one example of an inherent regularization scheme is SVM. So support vector machine has this nice C parameter, which can be used to weight error in its estimation process. So when C is large, you tend towards the hard margin, and it penalizes error points more strongly. So this results in a smaller margin, and when you have a smaller margin, there's typically more support vectors. There's more points that are influencing the selection of the parameters. But one of the things that's kind of interesting is there's no one C parameter that universally fits the data, that universally fits all data. So this is a figure from Hasty, 2013. He shows that with different radial basis kernels, so the width, if you want to think of it that way, or the sphere, the size of the sphere, has different performances based on the C parameter. So in some cases, certain radial kernels would prefer to have more regularization, but then when you have smaller kernels, maybe less regularization is better. So there's multiple parameters and things that should all be tuned simultaneously, if that makes sense. And so radial basis functions, I just put this as an aside, the size of the kernel might be somewhat analogous to, say, the size of a searchlight radius. Okay, so coming back to this question, is feature selection important? There's this really interesting paper by Carlton Chu where he had the ADNI data, which was trying to predict MCI versus Alzheimer's disease, so mild cognitive impairment versus Alzheimer's disease, just using structural MRI data. And he tried out t-test filtering as well as support vector machine recursive feature elimination. So wrapping a support vector machine within that iterative loop that I spoke about. And he came to the conclusion that feature selection methods actually didn't improve accuracy unless you were using some sort of a priori knowledge, like say using just the voxels in the hippocampus or something along these lines for trying to predict Alzheimer's disease or not. There we go. But if you take a closer look at his data, I think it's actually kind of interesting. This black line here represents the classification accuracy if you're using all of the voxels in the brain. And these other curves represent the accuracy for different variations on the t-test filtering or the support vector machine recursive feature elimination. And so I find this interesting because he came to the conclusion that feature selection didn't really help because technically using all the voxels did overall achieve the best accuracy. But if you overlay a 95% confidence interval here, you'll see that these other approaches using, say, 6,000 voxels as opposed to maybe 100,000 performed almost as well and within the 95% confidence range. So this is kind of, to me, a little bit of a theoretical question. If you can perform almost as well using a small fraction of the features, should you choose that option, or should you go with something more complex using 100,000 voxels, say? I'm not sure, but I guess because I kind of come from the parsimony camp, that the Occam's razor approach is what you would choose. So you should choose the model that's more simple, that with trading off just a small little bit of loss in the prediction capability. But it also depends what your purpose is. If you're actually just trying to make this diagnostic prediction, maybe you really do care more about the accuracy, so you'd rather just have everything, all of the voxels in there. So this is another example, another thing that I participated in, which was the ADHD 200 machine learning competition. This relates to feature selection, and to me, an extremely frustrating way. So in 2012, there was this public release of about 1,000 different structural MRI, functional MRI, as well as gender, IQ, all of these different parameters. And the goal was to take this training data and then to predict on 200 new subjects the ADHD diagnosis using these neuroimaging features. And so I was on the UCLA Yale team, and we placed third using tons and tons and tons of features. We started off with something like 100,000, more than 100,000, hundreds and hundreds of thousands of features, and we used SVM RFE, starting with all the features and kind of using a backward approach, trying to find the least amount of features that we needed. And we had resting state functional connectivity features, graph theoretic measures, ICAs, we had free surfer volumes, and it turns out that the winning team used only the demographic features. So this was extremely frustrating because they didn't use any neuroimaging features. So in theory, if your feature selection approach worked really, really well, if the only thing that was informative was gender and IQ and whether they were diagnosed in Beijing versus New York, then theoretically the feature selection algorithms should have just eliminated all of the brain data and found just those demographic features. But all of the teams that used neuroimaging features performed worse than the team that just used those demographic features. It still bothers me. I still haven't gotten over this. So I still talk about this because it still bothers me, and it really does, to me, still highlight why feature selection is definitely important however you decide to implement it, it's definitely an important step, especially because as neuroimagers, we have such massive data. We really need to think about how to pare that down into something that's useful for prediction. Okay, so I've talked a little bit about these hyperparameters, and maybe if you're using a kernel, maybe you have the width of your radial basis function. But how do we select these internal parameters given that we have a lot of different moving parts? Well, one way to do it is not just cross-validation, which you've already heard about from Jesse, but you can do something called nested cross-validation, which I mentioned earlier. So in this case, if you have a training set and then your out-of-sample test set, you break your training set further down into more training and testing sets, and you can loop through the training and test sets this way. So you can learn your hyperparameters, the parameters within your algorithm, using this nested approach, and then eventually apply it to the out-of-sample left-out test data. So this is just an example of how you can do different folds. So this is, I guess, a 2x3 or a 3x2. I'm not sure exactly how you would call this, but this is where you have 3 and you kind of iterate the first 2, and at each point, different groups of the data become the left-out test set. At some point, though, you might start to wonder, how do I know if I'm peaking? How do I know if maybe somehow in my code there was an error and it snuck in and I've accidentally been using some part of my training data on my test data with all of these different nested cross-validation? So some thoughts that come up, and this slide is from Nico Kriegeskorte, is some examples of ways that you might accidentally be doing this. The first here is that the same data is used for training and testing. Luckily, according to him, his subjective assessment is that this is very rare. But if you do this, then the severity is obviously quite bad if you're training and testing on the exact same data. If you have non-independent data used for training and testing, that's also fairly similar. This one here is a little bit tricky. So if all of your data is used to define an ROI, and then that ROI is then used as essentially as a feature selection approach, that's also peaking. If you set averages of analyzed data on the same data used for sorting voxels, that's also, I don't know, I think less common. And there's these couple others I'll let you read. But these are things to think about, different ways that your analyses can be circular. So you don't want to do that. So one of the things that I think is a good sanity check is to do a permutation test. So I have no idea why you would want to classify the difference between emus and pairs. But if you did, then maybe you would want to run a permutation test where you take the labels and you shuffle them at the beginning, and you hopefully get what you would expect chance to be. So if you're doing a binary task like this, and you shuffle the labels, and then you get 90% accuracy, then there's probably something wrong with your process. If you shuffle the labels and get roughly what you would expect chance to be, then you're probably doing good. I've also do maybe 10,000 permutation distribution, a null distribution of what you would expect the chance to be. And so I've also seen some people use that null distribution in ways to extract p-values from classification tests. So that's also something that you can consider. Okay. So thinking a little bit beyond just SVM, why would we want to do that? So as I mentioned before, each classifier has its own inductive bias. And so there's also this amazing theory called the no free lunch theory, which is actually not true at NITP. We all have free lunch every single day here. So aside from NITP, there's this Wolpert and McGreedy theory, which says they proved that no one classifier universally works best across all domains. And so it's a good idea to test out different models. This is an example of this is an example that I actually have from a few years ago on some human decision-making data. And it actually turned out, this is the group level result. I compared K star, which is similar to K nearest neighbor, but it's more stochastic, naive Bayes, support vector machine, decision tree, Ada boost, and random forest, which is a way of bagging different decision trees. And it turned out that SVM performed the worst. Linear SVM performed the worst, even after tuning the C parameter. So SVM is not always going to work the best. So the data that I was speaking of actually came from Sam Harris in 2008. He had people make these categorical judgments on things like, I own a toaster oven, and then they would have to say yes or no, I don't own a toaster oven, or mathematical or religious statements. And so that data was trying to make predictions about his data, belief versus disbelief. So one of the things that I thought was kind of fun, just as an aside, is that the decision tree-based classifiers worked the best for modeling human decision making. So here's an example of one of the decision trees, and this was using ICA-based features. So here's an example of the overlap between the ICA-based features that were used in this particular decision tree with the general linear model results. So I'm not actually going to do a formal two truths and a lie like some of the other people have done, but if I were going to do two truths and a lie, one of them would definitely be related to this. This would be my outlandish truth that I would hope you would choose as the lie, but it was actually true. After doing this study on decision making in the brain and being able to predict not just mathematical but also religious belief versus disbelief, there's this guy named Harold Camping who came along, and he predicted that the rapture was going to take place on May 21st, 2011. I think there was another one in 2012 and another one later. And so this group of British documentary filmmakers decided to follow his followers, and they brought some of them to UCLA and asked us to see if we could make predictions about their belief system. And so this is a bizarre decision tree. Do you believe Harold Camping's right? No. Well, I guess that's the end of the decision tree. Yes. Well, then does the rapture happen? If it does, then if you're a Harold Camping follower, then I guess you're taken up into the sky. If not, then I knew of people in Los Angeles that were planning to go on a looting spree and steal lots of shake weights. So, so anyways, this is a ridiculous truth. This actually happened. I actually did scan the Harold Camping followers. Unfortunately, nobody was able to come back after the May 12th. I kept asking them, can I pencil you in? Oh, but I won't be here. So they weren't able to come back for the second brain scan. They didn't think they would be here, so the scheduling was irrelevant. So another set of learning algorithms that are becoming quite popular is a lot of work from Nico Kriegeskorte on deep learning and neural nets. So neural nets can be shallow, they can have many hidden layers, they can have lots of nodes, they can be feed forward, feedback, they can be recurrent. The recurrent ones, some people say they argue that they're quite similar to Markov chains. So this is a whole other class of learning algorithms. Interestingly, a lot of the neural networks that I've seen have been winning the Kaggle machine learning contest lately, so it's worth noting that these machine learning algorithms do seem to do really well. And this is one of his results where they compared lots of different types of methods for estimating the machine learning algorithms, and these deep convolutional neural nets seem to, with many, many layers, seem to perform very well. But of course, there's this problem of what does this mean? How do we make sense of this from how do we learn anything about neural computation from machine learning neural nets, which were in fact originally inspired by the architecture of the brain? Is there any way to map this back to something that can help inform our knowledge about cognitive processes? So one of the unfortunate things about both decision trees and neural nets is that if you give them both enough parameters, if you give enough splits for a decision tree or just enough hidden layers in a neural net, you can actually learn any training data perfectly. So unfortunately, what begins to happen is once you've learned that training data perfectly, at some point you stop learning the information and you start to train on the noise. So this is overfitting. And so with decision trees, there's these internal parameters, like I talked about, that can help you do things called pruning, where you start with, there's two ways to do it. You can start with nothing and build upwards, or you can start with building a decision tree that classifies perfectly on the training data, and then you knock out randomly different nodes and you see how the error changes. And you have different thresholds of error tolerance for keeping those nodes. There's similar things with neural nets. You can do all kinds of different things with adding new nodes or adding new layers, and there's things called weight decay and weight sharing that can help you figure out if you want to add a new node or a new layer. So if you want to build upwards or if you want to start with everything and move backwards. So I really like this Alpayden book on machine learning. It's really a nice introduction and it's very readable. He says that you should always start with something complex and then add complexity. So if you start with something small and add complexity, then you can see if that complexity is warranted. I think had I done with this with the ADHD 200 machine learning competition, maybe we would have done better because we would have perhaps started with all of the demographic features and then realized that by adding the neuroimaging data, we were not getting any more traction, rather than starting with all of the features and progressively eliminating them. So it's just something to consider. Do you start with everything and move backwards, or start small and add? So sometimes you end up with the same result, but sometimes you don't. So just to summarize, this is kind of bringing back this thought from Russ Poldrak's lecture, where he showed the number of publications increasing and asked, with more publications, will we learn more? If we continue on this same trend where brain imaging and machine learning are starting to converge, do you think that we'll learn more about neural computation from machine learning? I'm not sure. I think it's kind of an open question. Hopefully, yes. Another thing to think about is that each time we train a classifier, it's usually that we're giving subjects a particular task, and that's the training data. And the test data is also that same task, almost always. And so on some level, if you have a really complicated neural net that's learned really well on the training data, the test data probably has a lot of overlap and is very similar. So in theory, it's going to apply very, very well. But how well will that neural net that you've just trained work on a new task? So in that sense, we're feeding our classifiers with each experiment data that's biased towards that specific task. And so I kind of wonder how well we can actually learn about learning, how well models from different tasks can be applied to other tasks, and if there are sort of canonical learning motifs, if those can translate across tasks. So one of the things that I find interesting is in machine learning, they have this thing called domain adaptation, which is a way of reweighting your model to fit new data. And there's all different strategies for doing this. And I thought it was interesting. There's this fairly recent paper by this group, Anderson, where they essentially did do this. I don't recall that they used this term domain adaptation, but they did take new semantic words and try to predict the brain the patterns of activity for that particular word by using a reweighting strategy. But in order to do this, you have to have a really nice model of your stimulus. So you have to have a model of your stimulus and how that relates to the stimuli that the model has been exposed to. So thank you all very much. Just very quickly, I wanted to give a quick plug for this toolbox called Weka. It's my favorite machine learning toolbox. It has a really cute bird that cannot fly, but it tries really hard to fly while you're classifying. And there's all of these lectures from this group. They have a Coursera and an MIT edX. So there's this amazing group of New Zealanders. They're funded by Google, and they have this repository of machine learning algorithms. They have tons and tons and tons of feature selection techniques, tons of dimension reduction, every algorithm you can think of, support vectors, tons of neural nets. It's huge amounts of decision trees. It's really amazing, and they have a really nice procedure for vetting the algorithms before they get added to the repository. So the machine learning community kind of vets the algorithm for a while before it eventually gets added. Doing things like nested cross-validation, it's just a simple flag. So a lot of these optimization procedures are really easy. And I worked with them over the past year, and finally now you can add your nifty data directly to Weka. So now there's this brain imaging toolbox. And I've demoed Weka in past years here at NITP. So if you're curious about how it works on just simple data, you can try out some of those demos and see the really cute Weka bird fly around. And if you're interested in learning more about the brain imaging toolbox, there's a whole set of lectures that are free online. So thank you.
I'll actually go ahead and relink Pamela's demos to our schedule for this year so you can see those. Weka is fun, and Weka is it's distinctly usable in ways that the MATLAB-based ones are distinctly difficult. Questions for Pamela?
Thank you very much for the great talk, Pamela. ADHD result that you weren't too happy with. I was just wondering, could that be due to the fact that the diagnosis to begin with is made mainly based on behavioral and observational data, so that actually performs a lot better, but once you bring in the brain imaging more biological data, it doesn't classify as well, because what we consider ground truth is based on something else. But in addition to that, could there be other subpopulations of ADHD patients that we're not really aware of, because again, we're classifying based on behavioral data, not necessarily from the imaging side of things? It's
Yes.
more of a comment as opposed to a question.
Yes, yes, I completely agree. And so that's partly why I started to become interested in what the underlying dimensions of ADHD were. And so that's why I worked on this non-negative matrix factorization paper with Ariana Anderson, who she'll be speaking tomorrow, to try and understand exactly how many different inherent groups there were within the ADHD population based on the brain data. So we were curious if the brain data supported the fact that there were, say, 3 different presentations of ADHD. And it's difficult because, as you're saying, we don't really have a ground truth. So sometimes you can take these clusters that you find and see how the classification accuracy changes if you have more or less. Otherwise, you just have penalties for the number of clusters that you find. We found 6, for example, seemed to be a nice number, but there was more information if you had more different groups. So, yeah. So the question of whether ADHD is dimensional versus taxonomic is, we decided it was more dimensional based on this particular data, but it is very hard. Where do you draw the lines? And should those lines be drawn at all? I don't know. So at least between the different presentations, it's difficult.
So what is the data size of that computation data set?
What was the data size?
Yeah, so how?
I think, well, it was 973, I think, in total. 200 of those were the left out sample. For each subject, we were given resting states, fMRI, structural MRI, IQ, gender. Some of the data provided things like the SWAN score, but not all of them did. So.
Yeah, so when you construct your features, so let's say the structural MRI, then do you construct some higher level feature, or you just give them the intensity?
So one of the things that we found is that because it was so different across sites, we actually found that it was better to train classifiers on each site independently, and then we used a neural net for learning the information across sites and making the eventual prediction. But it was just too difficult trying to find a mapping between the different sites.
So I'm just imagining, so at the beginning, if you include so many features, then if you have more features than data size, then you just have no choice. You have to use a certain type of feature selection, right?
Right, yeah. So actually in the WECA demo, one of the old demos that I have on the website, it actually shows that with 13, I think you start off with 13 features, and a couple of them are highly correlated, so sort of somewhat redundant. So even with 13 versus, say, 9 features, you can actually see a pretty big difference in the classification accuracy. So if you think about the brain, how many very close redundant features we must have, it really does make you wonder how, to me, feature selection, it's very important, doing something to increase the sparsity. So, yeah. Yeah. I wouldn't mind a repeat of the question.
You want me to repeat it or her to repeat it? Okay. If you're primarily interested in not just classification accuracy, but how which brain regions or voxels are most informative for your task and figuring out the pattern of activity that allows for some task, but it's not a great practice to just interpret the weights that you get from SVM, what would you suggest using? What sort of method would you suggest using? Searchlight primarily, or?
I would say voxel-based encoding methods.
Okay.
Yeah. Or, I mean, if you're really just interested in that, maybe you just run a GLM. If you're not interested at all in prediction or prediction accuracy, because it is very difficult to. I don't know.
You're still interested in prediction accuracy, but in terms of not overall how high can your accuracy be, but specifically which regions have the most informative information?
So that's actually a really good question. I think I had a slide that I didn't keep. I wasn't sure if there would be enough time, but you can actually use, if you do it carefully, you can use encoding and decoding collaboratively. So you can do something like run a GLM analysis on the subset of your data, which is your training set. Say you get a couple of different regions of interest that you're interested in. You can do just a comparison of which one has the highest predictive accuracy to try and then determine which regions contain more class-specific information. So if you do something like visual face recognition, if the images are really similar, maybe V1 will be activated with a GLM task, but it might not actually contain that much class-specific information. That might be more IT or something like this. So that's kind of an example, but you can certainly run those separately to see which areas contain more diagnostic information. Oh yeah.
about the feature selection, I know, Torweger used penalized regression and PCA that can do feature selection automatically, and there are some elastic net and other penalized regression methods that actually do a very nice job of feature selection. So I wonder if you can comment on that, or
Yes, I mean, absolutely. You can use L1 norm, or you can use the elastic net, the L1, L2. That Broderson paper that I had here, they actually used an L0 norm to get a really, really sparse. So you can use different penalties to increase the sparsity. And I'm not sure which particular paper you're talking about, but if you use something like PCA, then your features are independent. So this issue of the shared covariance doesn't really become a problem. So you can actually just interpret those weights directly, I would think.
right, I think that's what pretty much what Torweger's group did with a little bit of penalization. But I mean, there are some papers using elastic net and other regularized regression approaches with different cost functions that can actually do a very nice job of feature selection.
Absolutely, yes. That's a good point. That's a really good point.
Thank you for your presentation. I think you're right in saying that we're all tempted to look at the weights and try to make something out of them. And it seems like perhaps there's no way to salvage them. But imagine that you have several classifications within the same feature space, and so you'll have different weights for each classification that you do for binary classification. Certainly doesn't get at the covariation problem, but doesn't it become, if you can plot different sets of weights for different classifications within the same set of voxels, does it minimize the problem? Can you start looking at perhaps patches of voxels that have more weight for certain classes? I mean, they all share information. So it's the same set of voxels. The information sharing is perhaps similar, but if you have different weights over different classifications, can that help somehow?
That's actually a really interesting problem. That's a really interesting point. So you're saying that if you were maybe doing something like a cross-validation, like a tenfold cross-validation, to look at the distribution of the weights, is that what you're saying, over multiple runs, say, to see if that can inform that may be an interesting approach. What I've seen is that the redundancy really is a big problem. So something that has a strong, that contains a lot of information, if it's next to another voxel, or even if it's far away from another voxel, if they're both included, and they both have class-specific information, but it's similar, then the weights for both of them is going to be decreased. And it gets worse as you have more and more redundancy. And so when you're looking at the weights, the noise, if it's just but the same thing happens with the noise as well. So if you have lots of redundant noise voxels that all can absorb this covariance in a useful way, then those weights also do decrease as well. So in that Halfa paper, when they say to take the inverse transpose, that works really nicely if you have just two features, 2 channels or something like this, or just a small number of channels. It's not going to be too much of a problem. It's when you have a lot of redundancy that you really see the weights trail off. I mean, you could just look at it's a difficult problem. How do you make sense of the feature weights? And hopefully there's some way to salvage it, but as of yet, I don't know. There's also things with that back projection when you take the inverse transpose. You're making some assumptions about the distribution of the noise in that sense. If the noise is not Gaussian, it also doesn't necessarily work.
Hi, I just had a quick question about kind of what you're using to determine or to judge the classification, and if you've ever looked at using something like the reproducibility of the weights or the stability of the weight maps and kind of what your thoughts on are kind of incorporating that along with accuracy.
Like I said, I think that's a really interesting thing to look at is the stability. I haven't personally looked at that. I'm not sure. I'm sure somebody has looked at that in the machine learning world. But the other thing is with machine learning, in that world, most people are just interested in the prediction and the accuracy. They're less interested in this interpretation problem that as neuroscientists we're more interested in what's going on that makes this work. So I don't think it comes up quite as often, but I think that is a really interesting thing to look at, the stability of the weights that you both have brought up. That's a really good point.
I'm wondering how much it makes sense to interpret these weights, even if the different features are independent, in the context of machine learning being a multivariate sort of approach, and that it's really the combination of all of the different features that create good classification.
Exactly. Yeah. So you nailed it. It's the combination that produces the accuracy. And it's because of the multivariate nature that they all, if they have shared covariance, when they interact together, that's what makes it very complicated, the interpretation. So, yeah, I think you summarized it very well there, almost in a comment. That's what adds to the complexity of the interpretation, even with a linear classifier. So.
And what's happening when the features are independent that make us then able to interpret the weights?
So,
if that can be said quickly, or
yes, yeah. So I think if you think of that geometric interpretation of that figure from alpha, if there's this data is kind of rotated a little bit, so that rotation is the covariance. So if there's no rotation, if there's nothing shared between the two, then the weights, the way that it ends up getting assigned is that if the two are orthogonal and the dot product is 0 and everything, then this ends up not being a problem at all. You can just directly interpret the weight. So, yeah, it's this rotation which can cause the W vector to have strange assignments. So, yeah. I don't know if that helps at all.
Others? So join me in thanking Dr. Douglas for this. So our afternoon, if you look at the schedule, has time for you guys to work on debate stuff as teams, which